and welcome to day 3. But really, it feels like day 10. Oh my God. It's been a lot. It's been a lot. It's been long, but it's been great. But it's been long, but it's been great. So thank you for still showing up here bright and early at 9 in the morning. So turn to the person beside you and say, I hope you're doing okay. And then turn to the person on your other side and say, stay strong. We're halfway through the week, and I feel like this is the time when people start to fade away. They just peter out. The energy starts depleting. But yesterday was another packed day. We had parallel sessions on ecology and biology, predator and prey dynamics, research approaches, shark depredation, engaging fishers, non-marine frontiers, and we also had the group photo. And if you've seen the preview in the WhatsApp group, there's hundreds of people in varying states of trying not to die in the heat. There are different facial expressions of people like that, or like that, or like this. But we'll look back at it one day in 4 years with fondness and affection and be like, I was part of that, right? So we'll appreciate it in the long run. We also had student night last night. It was at this venue called Rise Up. And there were academic students, and there were students of life. And there was a very competitive trivia night. I don't usually get nervous, but then when there are shark scientists protesting on the answers and the questions, I start sweating. But then there was this group that won the very first trivia night for Sharks International, and their team name was Under the Finfluence. Are they here or are they hungover somewhere? Yeah? Okay, I think some of them are here. So I hope you've also been taking time to explore Colombo. If you read the General Information Guide, there's a line there that says pedestrian lanes are decorative. But in the last few days, I've also learned that stoplights are also decorative, and the lane lines and the flow of traffic are just suggestions. But today is a new day to learn and meet fellow shark and ray and chimera lovers. So if you've noticed, the password of Sharks International is Race Rule. And I think that's the organizer's way of showing love to the race, because maybe they think all the sharks or the true sharks are getting all the love. And today we have an entire session called Manta Matters, and we're starting today with a pair of keynotes talking about collaborative science for conservation action. I think my biases are showing. I didn't mean to say shark. So we have two amazing speakers. We have Guy Stevens and Marta Palacios. The first thing you will notice about Guy when you meet him is that he is very, very tall. But if you get a chance to meet him, you will feel his passion for manta rays. And it's that passion that led him to be the founder and CEO of Manta Trust, which is a UK-registered charity dedicated to the conservation of manta and devil rays, with collaborative projects in over 25 countries. He's the author of 3 books and over 50 scientific articles on Mobulids. I've also learned that there's another topic he can talk about with the same level of enthusiasm, which is birds. So I think Guy likes animals that do this in the air and underwater. Our second speaker is Marta. Her PhD research on Mobulid ray ecology and conservation along Mexico's Pacific coast led her to co-found Mobula Conservation. She is part of the ISRA, or the Important Shark and Ray Areas team, and has collaborated with the Manta Trust to investigate the global trade in Mobulid meat and gill plates. Something that Marta can talk about with the same level of enthusiasm as Mobulids is surfing. So, bringing the manta ray of sunshine this Wednesday morning, let's welcome Guy and Marta on stage.
How tall I am, I have to lift these up. Well, good morning, everyone. Thank you, Anna, and thank you to Daniel and all of his team for organizing such a wonderful conference. The last 3 days have been hectic. I've bumped into so many people I've known for so many years, and it's wonderful to have so many positive and collaborative conversations about helping to further the conservation of manta and devil rays. And that's what I'm going to talk to you today about, a bit about myself, the history of the Manta Trust, and some examples of the work that we do. And I get asked this question a lot. Why manta rays? What is it about these animals that, that has captivated you, that's drawn you into the last 20 years of my life studying these animals? And it's a hard question to answer in some ways, and I wish that the easiest solution would be to just take the person who asked the question into the ocean to spend time with these animals, because there's something extremely awe-inspiring about them, something captivating. I feel like they draw people through the looking glass into the oceans, captivating them and making them understand what is at stake if we choose not to protect our natural heritage. For me, these are animals I've been fascinated about for the last 2 decades, and I've dedicated my life to trying to conserve them and using them as an emblematic flagship species to try to engage people in ocean conservation more broadly. And this fascination started after I graduated from the University of Plymouth in 2003. I got a job working for the Four Seasons Resorts in the Maldives. I was amazed that they gave me the job. I was working on a luxury liveaboard vessel and traveling around the country, diving and snorkeling with wildlife, getting paid to do it. And literally week 1, you can see me here diving with a manta ray, and I was stunned at how amazing these creatures were. And then I quickly realized that you could also identify every individual using the spot patterns on its belly. And so I started to collect the individuals in a database, and I also started to think about whether I could turn what was a sort of a glorified snorkel guiding job into an actual research program. So I approached the resort management. They were opening a new resort about 100 kilometers to the north, and I said, hey, wouldn't it be great if you had a manta expert in the team and a project? And they said yes, and to my eternal gratitude, the general manager supported the idea, and for the last 20 years, the resort has been continuously supporting a research program in the country. And when I first moved to Bar Atoll, which is sort of central north in the Republic of Maldives, I wasn't sure that it was the best place to be to study manta rays, because I'd been traveling around the country and diving and snorkeling in other places that I thought were much better for these animals. But within a few months of being there, speaking to some of my Maldivian colleagues, they told me there was this special place called Hanif Fadzli, and it's not really a bay, but we call it Hanif Fadzli Bay. This is cul-de-sac of reef in very shallow water, and if you can probably see at the bottom here a couple of manta rays. They aggregate in this bay for foraging for several hours a day for about half of the year during the southwest monsoon. And it truly is a magical place. You can have aggregations of up to 250 animals all together in a very confined space, feeding for several hours at a time. So jackpot, I couldn't believe my luck having this amazing aggregation site. So I set about researching the animals. I grew the program. I got funding from Save Our Seas and some other organizations. I was able to increase the number of personnel. We started doing masters and PhD programs, and it's great. The animals seemed to be safe. It was really interesting behavioral ecology research. But then, after several years, the program had expanded to include hundreds of interns, and today the charity supports about 15 full-time staff, and they're spread throughout the country in different resorts and working in local communities, and several of them are here at the conference, so please do go and speak to them if you want to learn more about the program in the country. But then I took some trips to Sri Lanka and was exposed to something completely different. It was actually not far from here at Negombo Fish Market. I was writing a book with Thomas Peshak, the National Geographic photographer, and Tom and I were getting up early every morning for a week or two, going to the fish markets, and it was just pile after pile of dead manta rays and Mobulid rays. And I realized that everything wasn't so rosy for these animals, and there needed to really be efforts to try to address these fisheries, which were to some extent being driven by the demand for their gill plates. So there's bycatch, there's targeted fisheries in some places around the world, and unfortunately, as I spoke to colleagues, I realized that this was a problem that was much broader than just Sri Lanka. And so over beers one night, Tom and I hatched a plan to create a charity. I already had the project in the Maldives, but that wasn't going to be large enough and focused enough to address these really big problems that we were starting to realize were present. And that's why I started the Manta Trust. And that was in 2011. And it's a UK registered charity. The focus is on conservation, and we do that through collaborative research education programs. And we're trying to address the threats and to do it in a very strategic way. Right back in 2014, actually, during Sharks International in South Africa, a group of us got together, including many of the people that are here in the room today, working through an IUCN grant to come up with the first strategic plan for thinking about how to address the threats that these animals were facing. And over the years, that's evolved through a variety of different iterations. Some have been external to the Manta Trust, so thinking globally across all organizations, how can we address the threats through a more science lens, bringing in more strategic policy focus, then looking into how we need to have a more education diversity strategy as well, right the way through to much more impact monitoring and adaptive management efforts that we're doing today. And come on, computer. Here we go. And this has led to the creation of our current 5-year plan. So what we've really tried to do is drill down into what the key threats are, and then to work backwards from that to try to provide all of the resources and funding and strategic oversight to address these threats. Not surprisingly, the first two biggest ones are focused on fisheries, and I'll talk more about that later. But of course, there are other threats to these species as well. And all of this work is underpinned by importance of governance. Because we're a UK charity, I have a board of trustees. We have advisory board, we have structures in place to really ensure that we think not just through, okay, these are the outputs that we want to see, but how is we going to achieve that? Is the funding in place? Is there a strategic plan to ensure that we do that correctly and that we work efficiently and effectively to achieve these goals in a timely manner? And I would be remiss not to say that the Manta Trust is an umbrella organization. It's operated by a core team of about 15, but we rely on our collaborations heavily. We have, at the moment, about 32 affiliate projects, independent organizations, and you're going to hear from Marta in a minute, that we work with all around the world in Mobulid range states. And some of these projects focus more on education. Some of them, like Daniel's, focus heavily on fisheries management, as well as obviously policy. And so it's a combination of trying to address our big global strategy and working with our affiliates to help them, and obviously them help us to work towards these goals. So it's a constant collaborative back and forth. We can help our affiliates in a number of ways. We provide lots of assistance through whether that's fundraising, so directly giving grants or helping to raise grants for our affiliates and obviously for the central charity. We have several full-time fundraising team members. For those of you that have been here, might have gone to Jazz's talk on media and comms. We have huge education and outreach programs as well, scientific expertise, and help with strategic planning, etc. So we try to provide that sort of expertise in personnel at a high level that many of our affiliates don't have the actual staffing or capacity within their organizations. Because we're a registered organization, actually not just in the UK, the Madge Trust has sort of satellite registered foundations in Australia and the US. I mentioned the Maldives. And so that enables us to channel funding through sources of revenue that wouldn't be possible for our affiliates, and then we can pass that money through to them. So we can also assist with just the practicalities of moving money around in a world that often has barriers in that kind of area. It's also important to have a brand. The Manta Trust is this platform through which your voice can be amplified. So we can help with directly putting in place the media content, the websites, etc. But we can also amplify messages and have a clarity to cut through the noise to ensure that what we're trying to achieve is received to the broadest and the most targeted group of people possible. And obviously events like this, we can help raise funds to bring our affiliates, and we can also support through documentary making, filmmaking, etc. And then lastly, on the right, we have a wide number of resources that we make available. I'll just give a plug out to those. At lunchtime today, we'll be having a session on this new guidebook here. It's available in multiple languages. So anyone that's interested, who wants to learn more about how we actually have gathered together a lot of the information in a way that will enable people working in fish markets and landings, or people that are out in the ocean who want to collect observational data on these species. I've tried to condense 20 years of knowledge into a guidebook that hopefully is useful for that purpose. So we have lots of resources, codes of conduct, data collection protocols, etc., and a number of different portals online as well. There's education portals, so our education team put lots of effort into creating materials for all different age groups, right the way from young primary school kids up to university students. Our research portal, this is where we has a repository for all of the publications that the Madge Trust and our affiliates have produced. We try to make everything open access. So where possible, all of the information that's published in the literature can be made available to the public without being behind a paywall. It's also distilled into really wonderful, easy to digest infographics and other forms of more brief communications for the public. And then on the left we have Manta-Base, which is a new platform that we're rolling out at the moment. At the moment it's in Japan and the Maldives, and it contains these huge databases of photo ID sightings around the world. So the Maldives alone has like 100,000 sightings of about 7,000 different manta rays. And this is a huge resource. It enables researchers to go in and look at a whole variety of different topics, whether that's reproductive ecology or population size or threats. There's a lot of additional information that's tied to all of these sightings, and we make this platform available. It's got integrated photo ID recognition technology, and it's something that we hope will engage the public as well. Everyone can create their own accounts and track their own manta rays, and so it's a great way of engaging the public in the research, but providing a tool for the researchers as well. And this means that over the years, the charity has grown significantly. The graph just shows the number of personnel through the years. It started off with just me for the first 5 or 6 years, and then as the charity became established, we started to increase the number of personnel. We've also increased the amount of money that we can spend every year. We're still a relatively small charity. Last year, we gave out, or we spent £1.12 million. But we believe our impact is significant towards the conservation of these animals. And I have a short video to just highlight that. So that's a bit about the Manta Trust, how it got from my obsession with manta rays to this big organization. And being here at this conference and just seeing the amount of colleagues that there are present and looking ahead at the schedule, I know we're kind of dominating the next few hours in this room. I'm really obviously humbled and proud of what the organization has achieved. And I thought just for the second part of this presentation, I would talk you through sort of how the work that the organization has been doing. And I must stress that this is by no means a Manta Trust show. To achieve the kind of things I'm talking about here requires a massive commitment by hundreds of people working across dozens of different organizations, all with different expertise, different things that are essential to achieve these outputs. And ultimately, the governments of the world are the ones that make decisions at the high level, whether it comes to CMS or CITES. You'll also notice that I age considerably as we go through this presentation, and Daniel does too, so that's also good to know. So I wanted to talk a little bit about how we go from passion, research on the ground, to actually achieving some really important global policy measures. And right back at the beginning, the first thing I did was reach out to people like Sarah Fowler and other experts in policy and say, look, how do we get from all these dead animals to trying to put in place protections to regulate the trade? And just to go back again and clarify that mobulids, although they're fished and consumed locally, often caught as bycatch, which sometimes is discarded as well, there is a big demand for their gill plates, and those gill plates are traded internationally from the source countries to mostly Southeast Asia. And increasingly, their meat is also being traded across borders. So trade is a big part of the threats to these species. And if you're going to address trade, then CITES, which is the Convention on International Trade in Endangered Species, is the way to do that. So I said, well, hey, let's just push for appendix listing. Appendix 2 regulates trade. Let's try to go for that. But the people I spoke to said, well, look, hang on a minute. It's very difficult to get to that bar. And one of the first things you should try to do is jump over some other slightly lower hurdles beforehand. And they said, focus on CMS, which is the Convention on the Conservation of Migratory Species. And we agreed that the first approach would be best to focus on the mantas and not the devil rays. Not because the devil rays were less threatened, but we had less research. At the time, we didn't really have much data either on the mantas, but there was enough to do some IUCN Red List reassessments, moving the mantas to vulnerable status, and there was enough to put together a proposal. Mantas are much more charismatic. There was a lot more attention on them globally. There was also a global review of the economics, so the value of mantas in tourism, diving and snorkeling worldwide. Mary, who's in the middle of the picture here, led that study. And that was really important because it demonstrated an alternative livelihood for these animals in places where they're often exploited. So demonstrating that these animals are important and getting the public behind the campaign was also really, really valuable. And so in 2013 in Thailand at the CITES COP, the proposal was put forward and passed. And so amazing, manta rays are now listed under Appendix 2. And these COP cycles tend to go in 3-4 year cycles. So we then started thinking, okay, we need to gear this up to start focusing on the devil rays. So we went through the same process again, this time getting reef mantas and the devil rays added to the CMS appendices 1 and 2. Then moving through to CITES in 2016 in South Africa, which was also successful. And then we started thinking about, okay, we need to work more locally, national implementation, working with countries to put in place regulations to get these animals added to national protective lists. And then about 10 years go by, and we're working all around the world in fish markets, collecting data, and realizing that the trade is just carrying on as normal. And we realized that if we were going to move from Appendix 2 to 1, which would mean a complete ban on commercial international trade, we really needed to have strong evidence. It's a much higher bar to get from Appendix 2 to 1. And so we got the funding and we set about trying to conduct a global study to review the status of Mobulid trade. And Marta, who you're going to hear from next, was leading this study with a number of other colleagues within the Manta Trust. And we reached out to our huge network. We conducted lots and lots of interviews across 74 or so countries. We got together as much data as we could, and we were able to demonstrate that not only has the trade continued, it's increased, it's expanded, the meat trade is occurring in a much greater quantity than we realized. And clearly, the Appendix 2 listing is not working. The trade that we were reporting was completely outmatched by the trade that's being reported to the FAO and the CITES trade databases. So clearly there's a mismatch between what's being reported versus what's happening on the ground. And we also conducted market surveys in the retail markets to also demonstrate that there's a lot of product still being sold online in the source countries, in the sync countries as well. So that was the first global review that we undertook. And concurrently, we were also working on a global review of Mobulid fisheries. So this took even more effort across an even bigger team. A huge amount of work went into gathering of this information. This is the big review paper, but there was about a dozen other papers focused more on national efforts to publish on the declines that we were observing as well. So trying to characterize the fishery, what different forms of gear are being used to catch these animals, how they're being landed, where they're being landed, what species, in what numbers. And the total annual catch for Mobulids currently is about 250,000 individuals a year. And unfortunately, this is leading to massive declines in their populations. Each line is a different species in a different population. And across all ocean basins, across all species, there is steep declines in these animals, and the declines are accelerating across time as well. So clearly, there's really significant fisheries pressure, there's continued trade, and this information was then used, along with lots of other research by a load of other research groups as well, to contribute towards CITES Red List reassessments for the 3 oceanic devil rays, and that was done late last year, and all 3 moved now to critically endangered, and we'll be working on the remaining species in the coming years with Rima and her team at the SSG. And so that information went into a proposal to have these animals moved to Appendix 1, and that was put forward by the government of Ecuador last year at the COP in Uzbekistan, and amazingly, wonderfully, these animals are now all listed under Appendix 1. So we've seen increases in protections, which is really great. However, we're seeing these animals all move up the threat status as well. So we clearly have lots of work to do. While it's important to achieve these high-level policy gains, it doesn't mean that these animals are protected. And so now we have to pivot to or expand our work on national implementation, species management plans, regional action plans, enforcement capacity, so traceability, what's happening to the ongoing trade. Is it legal where it's illegal? How is it getting through the regulatory frameworks? And then working with the fisheries to try to regulate the bycatch, try to reduce it and reduce mortality. So lots of effort and future work needed. And I want to finish by saying that would not be possible without this massive team that we have here and across the world. And thank you to my wonderful staff and to my network of affiliates and colleagues, and of course to the funders. None of this would be possible without their support. So with that, I will say thank you very much, and I will pass over to Marta.
Hi, everybody. It's so nice to put faces to all the people that I've been chasing with many emails for the global trade, but also for the Israel staff. So just thank you for being here, and also thank you for Daniel and all the organizing staff for this amazing conference. Today, I want to introduce you a little bit of the work that we do in Mexico and take you to a journey to the global conservation of sharks and rays that is currently happening now in the world. So 10 years studying Manx devil ray taught me this lesson, that strength comes in numbers, and sometimes in groups of 17,000, like this group right here. Manx devil ray of the same size school together. And just like them, we are strongest when we collaborate at all levels, but especially when we collaborate with those at our own level. Shark and ray research should be one of the most collaborative fields in marine biology, not only because of the global distribution of many of these species, but also because of the migratory nature and the need to work across diverse stakeholders, from fisheries to tour operators, divers, governmental agencies, and institutional international bodies. Normally, scientists collaborate at large scales between NGOs, senior researchers across countries, and professors within labs with their students. But yet, as a student or as an early career researcher, you might feel like this cow-nose ray in the middle of the Manx devil ray school. Your place in this collaborative network can feel a little bit uncertain and sometimes frustrating, because you have a lot of ideas for conservation solutions, but turning them into projects seems sometimes impossible, mainly because of the lack of funding. So today, I want to share with you 3 examples of how collaborative science with scientists at your same career stage can help not only accelerate the study of lesser-known species, but also help map critical habitats for sharks and rays around the world. So, although I'm originally from Spain, I spent the last 10 years in Mexico, where I conducted most of my university studies, from my bachelor through to my PhD. And I have to confess that, unlike many of the people in this room, I was not born with a passion, an early passion for sharks and rays, but I always been fascinated by nature and animals, especially for those that make you stop and think like, what on earth is that? So that curiosity took me to work and volunteer in the study of, unlike less common species like the yellow-eyed penguin in New Zealand, the pink river dolphin in the middle of the Amazon, and finally oceanic manta rays in the Galapagos Island. While I was on this project with Manta Trust, I realized very soon that there was a lot of information on the bigger species of mantas, but very few information was available for the smaller species, the devil ray. So in search of this unusual aspect of the animals, I decided to move back to Mexico and start a project studying one of the lesser-known species of devil rays. So this is the Manx devil ray. It's one of the 4 species of pygmy devil rays, is catalogued as vulnerable, and is distributed in the Eastern tropical Pacific. It reached 1 meter and 30 centimeters, more or less, and at that time, in Mexico, in the Gulf of California, specifically at the Espiritu Santo archipelago, there was an aggregation of these animals that were attracted by the zooplankton, concentrated by lights. And a small diving activity was already taking place. So in 2016, this seemed like the only place where you can find these animals in a predictable and repeatedly way. So I decided that this looked like the most promising place, because in Mexico since 2007, mobulids have been protected. So all the fisheries data disappear, and they are no longer available at landing sites. They are also too small to be tagged while diving, and they are more difficult to catch than sharks. So you might think that this all had the ingredients for a monumental disaster because I started this project that nobody was kind of interested in, just very few, and of course nobody had funding for. So yeah, it almost went wrong. But for the first 6 months, I was really struggling. I had no funding, no way to reach the island that was 30 kilometers away from the coast, and no real chance of capturing this species. So in a last desperate effort, my local supervisor sent me to a remote island in the middle of the Gulf of California, the island of El Pardito, where a family of fishers had been living there for more than 120 years, and where the professor Donald Kroll from the University of California has been working alongside artisanal fishers to study turtles and some other Mobulid ray species in the Gulf of California. So I was sent there during the summers, and I was persistent enough to convince Donald Kroll to help me with the project. So he helped secure a $5,000 grant, and more importantly, he introduced me to Juan and Felipe Cuevas, two of the fishers that knew how to capture these animals with the rodeo technique, which is a special technique that allows to capture mobulas not only when they are moving, but also when they are in deeper waters. At the same time, at my postgrad center, I met Abel Trejo, who at that time was finishing his master on nursery areas in La Paz, and luckily for me, he was more than happy to help. So soon I realized that these three persons were like the most important assets of my project. As we enter the data collection mode, we wanted to understand the habitat use of this species in this archipelago in the Gulf of California. We started gathering all the data we could to maximize our time and our limited budget in the island. Abel, at the same time, was teaching me all these methodologies that he had learned in his master's. We were doing acoustic telemetry, traditional tagging. We were collecting blood samples to do isotopes. We were also doing zooplankton tows and underwater visual census. So I was exploiting them. I have them working for 15 hours a day, 5 days per month, every 2 other months for a year and a half. And after that, we were able to describe for the first time pygmy devil ray nursery area, where neonates and young of the year stay for up to 8 months, while the adults come in spring and summer to mate and give birth. Because this area lies within a marine protected area, my project involved a lot of visits to the CONAM, which is the Mexican Authority for Protected Areas, and there I not only applied for research permits, but I also introduced myself and present the project emphasizing the importance of this bay and why it needs better protection measures that they currently have at that time. However, I have to confess again that there were moments where I hesitate, not because the problems and the solutions were clear, but because I didn't feel like I have the authority to speak up for these animals. No, I was just a merely a student. However, I knew the problem, and I was the only one doing something. So at that time, responsibility mattered more than title, and I decided to speak to them. So this is how our collaboration with the Mexican government started. We then put together some code of conduct and management rules for this bay and for the better protection of this species. So this lesson taught me that sometimes you just have to start a collaboration, you just have to show up at someone's office in a very consistent way with ideas for solutions, but also with a willingness to engage. Abel actually taught me this during our fieldwork, where he was coming to every field trip, even in his 3 days. And where we were gathering all the data together, he was sharing his ideas, skills, and perspectives. And we had this, create this space where we were able to discuss and establish the methodology to collect the data based on our funding and the limited time frame that we had. So as students, we all confront limitations of budget, limited attention from our supervisors, and a lot of self-doubt, especially during our early years. But if we can canalize that frustration towards the collaboration with our peers, we will find that we have people that are eager to start putting science into practice and start doing, that has more time and a different and updated skills and more availability to discuss. So after my master's, I still wanted to do more for these species that were still facing a lot of threats. In the Gulf of California, we have a huge problem with the gillnet fisheries targeting stingrays that get amounts of bycatch that can go up to 80 animals in just one night. And also we have a new threat that is a growing and unregulated tourism to observe these animals. So I decided to pursue my PhD still with these animals to understand the critical habitats and the fisheries vulnerabilities for these species. But at that time, I knew better, and I knew this was too big for just one person. So that's when Mobula Conservation was born. It was co-founded by 2 PhD students. One of them was me, and a postdoctoral researcher, with the idea to create a collaborative space where science serves action to help mitigate the threats that these species are facing in the Eastern Tropical Pacific. So at the island of El Pardito, I met Melissa Cronin, that at that time was a PhD student at the University of California in the States, and Nerea Lecema from ASTI in Spain. They were also studying devil rays in the Eastern Tropical Pacific, and especially focusing on the topic of bycatch. We were, the 3 of us, 3 early career scientists working on the same species, in the same region, and asking the same questions. And instead of competing, we decided to collaborate. So, although this began as a kind of emotional group to survive all the ups and downs of a PhD life, it quickly evolved into a very collaborative and safe space where research support, ideas, funding, and publications were actively shared among each other, and where we became really involved in each other's project. And because each of us were based on a different university, on a different country, we all had access to different spaces, different platforms, and different data sets. So when we combined the 3 of us, we were able to multiply our opportunities. Also, although our collaboration was not based on funding, just on growing opportunities, our credibility and a stronger track record of the science that we were producing was bigger, so it was easier to access funding. And at the same time, because we were now kind of like a platform with more structure, it was easy for us to collaborate and establish bigger partnerships with NGOs such as Manta Trust. So, also combining 3 brains and 3 sets of skills helps to produce more science in a less amount of time. So in the past years, we've been able to publish more than 15 articles on these lesser-known species, addressing different topics as the ecology, that answer questions of where these animals are and what are they doing, to the threats they are facing, that goes from fisheries vulnerability to illegal trade, not only locally, but also at a global scale, and practical solutions on fisheries, especially industrial fisheries. So Melissa is going to talk about this today. So please go and check her presentation because it's really interesting. So yeah, after all this, Manx devil ray is now a little less unknown. And not just because I'm talking to you, 100 people in this room, but also because we've uncovered some of the basic notion of their ecology. We know now that they rely on shallow and protected bays as nursery areas in the Gulf of California, that they can form aggregations, seasonal aggregations of more than 17 individuals in a single group, which exacerbates their vulnerability to fisheries, that they segregate not only by size, but also by sex, and that pregnant females form distinct groups that also exacerbates the vulnerability to fisheries. We describe new reproductive behaviors and also expand the reproductive season from 3 months to 6 months during the spring and summer. In Mexico, we identify critical habitats such as reproductive grounds, but also migratory routes with animals that travel up to 500 kilometers in less than two weeks around the peninsula. We also learned that they shift the diet as they grow. First, they feed on euphasids, and when they are older, they feed on mysids, and that they are still sold in local markets in Baja California Peninsula under umbrella terms such as Manta raya and pescado seco. We were able to uncover other threats, like the gillnet fishery of stingrays is having a tremendous impact on this species, as I mentioned before. One fisher can get 80 mobulas in one night, which is blow-minded. And then we also learned that the post-release mortality of these animals, once they are being caught and released, is more than half of the individuals do not survive. We also learned that the other methodologies for mitigation strategies for bycatch, such as the green LED lights, do not work for this species and this fishery in particular. But there's also good news. So since 2019, there's been a growing tourist activity in Baja California Peninsula, in Mexico, that we estimated an economical value of $8.5 million million in 2023. So the local communities that stopped fishing for these animals 20 years ago due to national protections are now benefiting from the tourist activity with these observations. And today we are working with the Mexican authorities of Semarnat and 4 local communities to establish the science-based recommendations for this activity so it can be sustainable not only for the animals but also for the people who depend on them. So what started as a collaboration between a few early career researchers studying a couple of lesser-known species showing me something bigger, that this model works and that it can be escalated. So as Guy was explaining, in 2025 with Manta Trust, we were able to assess the global trade of mobile arrays around the world thanks to a global network of researchers and collaborators. So what if we could scale that even more to all the shark, ray, and chimera species and to all oceans of the world? I was very grateful that my journey took me to work in the important Shark and Ray Areas project led by the IUCN Shark Specialist Group for the past 2.5 years. And I believe this project might be familiar to many of you, as we've now covered more than, all oceans of the world with just the African Atlantic region left. So this process aimed to delineate critical habitats for sharks, rays, and chimera around the world that encompass the different life history functions of these species, and has brought together more than 1,450 contributors from around the world in person and online. So, we cannot solve global conservation problems with fragmented science. Our field depends on collaboration, but we don't always structure ourselves to enable it. Unlike many scientific processes, ISRAs, the important shark and ray areas, are established on a model that everyone contributes at the same level. If you have data and are willing to share it, doesn't matter if you are a diver, a fisher, a student, an early career researcher, or a senior scientist. We welcome everybody to participate, and that has enabled to get to this point. This approach showing me the true collaboration, the power of the true power of the collaboration among scientists, not only within the ISRA team, which makes an incredible job working too much, but we were able to identify almost 1,000 ISRAs around the world, encompassing 938 species, which is half of the shark and ray species that are described globally, covering almost 5 million kilometers of the ocean surface. So this work was impossible to be conducted by a single lab or country or data sets. It was only possible because of the global collaboration. And what makes me particularly happy is that some of the aggregation sites and the reproductive sites that we identify in Mexico are now on this e-atlas, and it has evolved from a few field researchers doing field work in a remote island in Mexico to now informing global conservation priorities. And all of this is publicly available on the e-atlas, enabling scientists, fisheries bodies, and governments to engage with the data and integrate it into spatial planning, but also management. So these areas provide the scientific foundation needed to move from knowing where the species are to understand how to better protect these areas. So we are here, and if there's one thing that I learned from this small island in the Gulf of California to a global network of collaboration, is just that collaboration is not just helpful, it's what makes conservation at a large scale work. So we are here, most of us studying pretty much the same species in different areas and asking different questions. So I really encourage you to start talking and trying to build these collaborations, not only with the people that you look up to, but also with the people that will grow with you in this journey. So I also wanted to thank you to the Mobulid conservation team that it's been growing for the past 6 years. We have now a lot of students that are doing an amazing job. They are also presenting here. And yeah, thank you all for being here.
Cool. Good morning, everybody. Hi, my name is Janis Argeswara. I am currently working at the Marine Megafauna Foundation in Indonesia. And today, I would like to start off this long Mobulid heavy talk with talking about reef manta rays in Indonesia. So Indonesia holds some of the world's most important reef manta ray aggregations in the world, And at MMF, we've been watching some of the sites for over a decade. But Indonesia is massive, and I only have 10 minutes, so I can't talk about everything. So I'm gonna talk about 2 of them. But before I go into the sites that I want to talk about, I would like to do a shout out to Dr. Edi Setiawan's and team's work up in the Raja Ampat MPA network, where they have been doing equally foundational long-term monitoring on population demographics and movement of reef manta rays up there. Now, I'd like you to focus on this little yellow square. This is called the Lesser Sunda Seascape. And it is in the Lesser Sunda Seascape that we are able to find 2 massive manta ray aggregations, one in the Nusa Penida Marine Protected Area and one in Komodo National Park. Now, I'm sure all of these names are quite familiar to you, as these are really heavy tourism sites, so a lot of tourism within the country, but also an international destination. And these 2 sites, these 2 sites are also have been identified as important shark and ray areas for reef manta rays, and I will tell you why they're identified as an ISRAS. So going to Nusa Penida MPA, we are now in Bali, and we have identified 2 critical habitats. One is Manta Point, which is a cleaning station and reproductive ground. So we have seen a lot of cleaning behavior of reef manta rays over there, a lot of really pregnant mamas, a lot of mating and courtship behavior as well. And the other one is in Manta Bay, where it is a feeding ground. So we have seen a lot of feeding behavior, very, very healthy and productive plankton abundance over there as well. And as you will see later, why it is a potential nursery ground. Now, since 2012 to 2023, we've identified over 800 individuals, 900 actually now, and with the resighting rates of almost 80%, which means the manta rays keep coming back there again and again and again, which kind of emphasizes why the Nusa Penida MPA is an important site to these reef manta rays. Now, focusing on Manta Bay, why is it a potential nursery ground? Well, visually, we have seen a lot of juvenile or immature male manta rays with unclassified claspers over there, and we have seen some neonates or manta rays that were just born recently with sac cord scarring on their bellies. But other than visual cues, we've also measured them using photogrammetry to try to get an accurate disc width estimates. So this is the data that we have right now, and I know the sample size for Manta Bay is very small. Please bear with me. We have collected a lot more data, but I just haven't had the chance to put it up here. I need to lock myself in a room to actually get that done. But even with this small sample size, you can also see the pattern initially, where in Manta Bay, most of the manta rays that's been identified are less than 3 meters from wingtip to wingtip, while the ones at Manta Point are generally on the larger size. Now, obviously, I need to do the analysis to actually get a better picture of the different use of the sites. But even with this, you can already tell the difference that different sizes of manta rays use different habitats. Now, talking about population, luckily, when we found out that population of manta rays in Nusa Penida is increasing, and this is using the capture mark recapture model. Using data from citizen science, so about 16% of our data come from mantamatcher.org, which is a global manta ray database. This graph tells us 2 things: that the annual population abundance is increasing, and that citizen science data are actually contributional to our data collection. And manta rays were protected in Indonesia in 2014, all across Indonesia's exclusive economic zone. And we believe that part of the story why the abundance is increasing is due to that protection. Now, moving on to the east, we are at Komodo National Park. And right now, it is the largest reef manta ray aggregations that has been documented in Indonesia. So far, we've identified over 1,300 individual manta rays. A lot of them have been seen cleaning, feeding, and there's also a potential nursery ground up there in the north site. And a lot of these manta rays are found across 4 main sites, two in the central site, central Komodo, 1 in the south and 1 in the north. And about the resighting rate is about 72%, so it's still quite high. Now, we've seen 2 large aggregations, 1 in Bali and 1 in Komodo, which is about 450 kilometers apart. The question is, are they actually connected? Well, I'd like you to meet Miss Caroline over here. So, before I talk about her, if some of you guys aren't familiar with reef manta rays, they actually have unique patterns on their bellies that acts as like fingerprints for us, and we call them belly prints. And so when we see repeated individuals, we can actually tell that it's the same individual. So here, Caroline was first seen in Komodo in 2016, and then in 2019 and 2021, she was seen in Nusa Penida, which is over there on the west side. And then in 2023, she was seen in West Sumbawa. And this tells us that the same individual, as you can see all the photo ID above there, actually travels across the Lesser Sunda seascape within 7 years. Now, she is not the only one. Studies have shown 14 other manta rays that actually move between Nusa Penida and Komodo National Park, although there's no distinct seasonality yet. Maybe there's just more dynamic of their movement. So we don't really know why they move, but we have known that a bunch of them actually move between aggregations. And satellite tagging from Dr. Eddie Setiawan's work also revealed that manta rays tagged in Komodo actually uses the corridor in South Sumbawa and West Flores, and the manta rays in Nusa Penida actually travel beyond MPA boundaries and also using Lombok and Southwest Sumbawa as a corridor. Now, while the satellite tagging shows that these manta rays move, but it haven't really shown that these manta rays move across aggregations and that they're actually together. And so satellite tagging got us partway there. Photo ID confirmed that manta rays are seen in both sites, and fins crossed acoustics tagging will actually close the gap. And this is the motivation behind the Lesser Sunda Seascape Tracking Network initiative. So some of our research priorities is we have acoustic telemetry, like I mentioned before, and we also want to look at reproductive ecology and life history studies, hopefully using ultrasound. And then to do threats assessment on anthropogenic threats and also climate-driven threats, as well as population modeling on a seascape level. And what is conservation if we don't talk about education and capacity building and outreach, where we have been using virtual reality to give students and local government figures access to the ocean. And so we want to emphasize that manta rays are livelihoods for a lot of people, and that protecting manta rays is a shared and collective responsibility across jurisdictions. So that is from me, and I'd like to thank everybody here, and that's outside of this picture. And thank you so much.
Thank you very much, Janis. No time for questions, I'm afraid. So moving quickly on to our next presenter, Luke Gordon is going to be talking about his work on drone surveys revealing oceanic manta ray show exceptionally high fidelity to a turbid coastal bay in Fiji. So welcome Luke to the stage.
Thank you, Guy. Oh, sorry. Okay. I've only got 10 minutes, so I'm going to crack straight into it. When we think of oceanic manta rays, we typically think of them being associated with offshore oligotrophic habitats. But in Fiji, we are showing that this species can actually occur in shallow, turbid coastal environments, and not just occur in those turbid environments, but actually show a high degree of site fidelity. Now, just to give you some background on oceanic mantas in Fiji, we had no concrete evidence that this species did occur in Fiji waters. But in 2018, we managed to find 2 individuals feeding off of the capital city, Suva, and we managed to jump in the water and gather the necessary footage we needed to confirm that the species did indeed occur in Fiji. Now, from that point onwards, we were pretty interested to find out more about these manta rays, so we started to conduct sporadic boat surveys in the bay. And over the next 3 years, we only had another 4 confirmed sightings of Mobulid. And interestingly, one of those sightings was of a manta ray we had seen in 2018. So 6 sightings of oceanics and 1 resighting already. Anyway, in November 2021, we went out with a videographer. We found a manta ray and we put up the drone, and that one manta ray was actually accompanied by another 6 individuals. So we obviously made it a priority to start drone surveys in the bay. And I just want to mention that our surveys were not strict transect-based abundance surveys. We were mainly focused on gathering individual identifications. But needless to say, the drone opened our eyes to a whole new world. Lathola Bay is one of our, I don't know what's happening there. Lathola Bay is one of the most incredible habitats we have in Fiji. These mantas are feeding in incredibly turbid conditions and very shallow conditions. They also somersault on the surface, which allows us to gather the necessary identification footage that we need. Now, to give you a bit of context about Lathola Bay, it sits in the southeast corner of Fiji's main island. And the bay, as you can see in the top right, is spatially not very big. Its widest point is roughly 8 kilometers across, and it only reaches 15 meters deep on average at high tide. It's very shallow. It is mainly defined by 2 aspects: Fiji's largest river in the east, the Rewa, and Fiji's largest city in the west, Suva. Now, the Rewa deposits large amounts of fresh water and sediment into the bay every day, and also from Suva, over 1/3 of Fiji's population lives in that corridor, and the sewage outflow from the city feeds into roughly 2 kilometers away from where these mantas are feeding. So you can just see from these photos how dynamic the bay is and some of these dynamic mixing zones that we see. Visibility ranges from pretty much 0 to, on a good day, we'll get 3 meters in this water, which obviously makes in-water work very hard, which is why drones have been so successful for us. Now, here you can see embossed on the map our survey area via drone. We survey from shore, from the university campus, and obviously we're limited by battery and signal to the remote controller. So the embossed area you see is our survey area, and the density is where we have seen our oceanic manta rays. So you can see it's quite a small spatial area in the bay. Now, what did we find? So from our raw sightings, we firstly saw that sightings looked like they were seasonal. Mantas would appear in May and disappear in December. Now, survey effort wasn't consistent throughout this time, but our binomial GAM confirmed that day of the year did have a significant effect on occurrence, and the peak was in September and October. And 96% of our sightings were of feeding behavior. And I just note, before I move on to the next slide, that we had 874 sightings recorded of a very small aggregation of manta rays. We only have 22 individuals in our database, 17 females and 5 males. Obviously, that means our sightings are heavily skewed towards the female side. We have 8 individuals that have been sighted over 50 times each, and we have 2 individuals that have been sighted over 90 times each. So we wanted to investigate site fidelity, and we did that through 2 ways. We looked at our resighting metrics, and we looked at lagged identification rates. We had a resighting rate of 95%, just to give you some comparison to other populations. It ranges for Byrostris from 12% through to 35% across different areas of the world. We had a mean recapture rate of 34 times per individual, and we had a multi-year recapture rate of 87%. Okay, so our data was showing that these mantas are showing inter- and intra-annual site fidelity. This was also confirmed by our LIR plot, that the decay doesn't reach 0 even after a 700-day lag. So we can see the probability of sighting a manta again was still high after 700 days. And residency. We investigated residency through 2 methods. One was fitting maximum likelihood models to our LIR data, and also through a residency index. Now, our model was a good ecological fit, and it spat out some figures for us. Mean residence time inside the bay of 53.6 days. Mean time outside the bay was only 8.7 days, and our residency index was 13.9%. Now, I do just want to make a note on the residency index. Most studies that create this index use acoustic telemetry. We're using photo ID studies, so do take this figure with, or interpret it with caution. However, we did use it to compare to the other 2 figures that have been reported for Byrostris, and it's comparable. In Indonesia, it's similar at 14% residency, and in Mexico, it's around 17% residency. And you can see our residency was different across individuals. Our binomial GAM showed that individual ID was a significant random effect, showing heterogeneity in our population. Now, we did fit a binomial GAM to our data and showed that occurrence was influenced seasonally and temporally and environmentally. But I'm going to just move on because I know I'm running out of time. But catch me after if you want to discuss any of these results. So, just some of our emerging insights. We are showing that you can dorsally identify oceanic manta rays. Each of our individuals has unique dorsal identification patterns. And at the moment for us, we're at 7 years stability on those. We also see a lot of microsympatry in the bay, which obviously asks the question of the plankton complexity that's going on inside these waters. And this guy is rearing his head at a lot of different sites, a suggested shrimp, the Lucifer species. And the bay, unfortunately, has very high pollution. We have high fecal coliform loads, we have high heavy metal pollution, we have high nutrient loading, and we have high pharmaceuticals. So these mantas have a bit of a situation to deal with. And just in conclusion, we've obviously revealed high site fidelity to a contrasting habitat that these mantas are normally seen. Individuals are showing strong inter- and intra-annual site fidelity, with residency being seasonal and not permanent. And also, I think the important point is this study corroborates mounting evidence of plasticity amongst the species, and that critical habitats might not be where you think they are, and they could be overlooked. So for us, it was in this human-impacted coastal bay. So finally, thank you to everyone who has supported our work, namely University of the South Pacific, the core Manta Trust team, and Dr. Mark Erdman. So thank you very much for listening. Thank you.
Thank you very much, Luke. We're still trying to catch up on time, so no time for questions. Next to the stage, I'd like to welcome Jess Haynes. Jess is going to be talking about her PhD work on the characterization of a nursery site for reef manta rays in the Maldives. Over to you, Jess.
Thank you very much. So it's a pleasure to be stood up here and be standing in front of you. I'm in the final year of my PhD, so I'm very much an early career researcher, but throughout the last few days, I've met my elasmobranch heroes, and I'm sure a lot of you in the room have as well. So it inspired me to acknowledge someone that is not here and never really will be attending these kind of conferences, and that's our boat crew. I think there are unsung heroes. We know that funding is needed to do research. We know that academic supervision is needed to publish papers, but these guys taught me everything I needed to know about where to find the mantas, where we should go if they're not where we thought they would be. And I really think this whole project I'm sharing with you today is with thanks to their knowledge of them growing up in that atoll. So I wanted to make sure they got a mention in this talk. So hands up if you have seen a baby manta ray, a live baby manta ray. Okay. Nice. Hands up if you've seen a dead or deceased young individual. Yeah. So from having worked in the Maldives for all of this work, I wanted to introduce you to Coco. We shouldn't have favorites, but he is my favorite manta ray. What he's doing right here is he's flipping upside down and very much looking at me on the surface as I am looking at him. These young of year are so charismatic and curious, and I might have been the first human he ever interacted with, but I will never forget this individual. In the Dhivehi language, Coco means little brother, so we thought it was quite fitting for this young male. So just to draw your attention, although I realize this is a very manta-heavy session, so you're going to be experts in biology by the end of today, we identify young of year by their small claspers, if they're a male individual, their small wingspan, particularly for the females, and their very light spot patterns, which stay the same throughout their lifespan, but do get darker as time goes on. So even though we don't know where they're born, specifically, we do encounter these young of year individuals. So that sparked the question while I was working in the Maldives, collecting data on these young of year that we were encountering. Okay, what is a nursery, and is this lagoon where I'm working a nursery for reef manta rays. So following the Hupel et al. Criteria, which was first developed for sharks in 2007 and developed for all elasmobranchs in 2019, we looked at the 3 criteria to run these kinds of assessments. So first, are they young of year and juveniles encountered more frequently in this lagoon compared to anywhere else? Do they remain in that area for long periods of time? And is that area repeatedly used over the years, or was it just a one-off phenomenon that they happened to like that area? So my study area is in the north kind of central area of Ra Atoll, which is the red box that you see in figure A. But the lagoon itself is highlighted by the orange boundary in Marmenegao Lagoon. So I lived on Marmenegao Island, which is a 5-star resort, the Intercontinental Hotel that are funders of the Manta Trust and host a year-round Manta project there, which allowed us to collect this kind of data. So for the study, we separated Marmenegao Lagoon into the inside reef, the fringing reef, and compared this with a secondary site, Femfushi, which is just north of that area. Unfortunately, Femfushi is really hard to get into. My field assistants are actually here today, and they will remember that we had a lot of difficulties. So unfortunately, the data is not that comparable. So we used the rest of Ra Atoll and the sites you see in white to do our comparisons for this study. In the Maldives, there's a nationwide ban on telemetry tagging. A lot more work is being done about community engagement, raising awareness, education to try and overcome this, but it meant that this study was very much based on using aerial drones to help find the animals, photographic identification to answer our questions, remote underwater video surveys to get extra data, leaving the cameras at the cleaning stations for many hours, and stereo video photogrammetry to measure the individuals. So our results showed that we had 2,946 sighting events, which is a manta seen on a certain day, of 355 individuals found inside Marmenegao Lagoon. It's mostly active during the northeast monsoon, and throughout our years 2019 to 2022, we've identified 45 young of the year. Might sound like a large number, but you have to remember that these individuals don't mature, sexually mature, until they're 14 years old for females and 11-year-old for males. So our job, and Joshua Stewart said this very well, that we need to understand more about the young life stage. We need to protect the young life stage if we're going to protect the population itself. So the smallest individual we measured is Coco, who was the one you saw earlier in the video, and he was measured at 163 centimeters. And there's an ongoing study by Neve Froman, who's also doing his PhD, which we took daily measurements of these individuals throughout the years, and he's looking at the growth rates of these young mantas. From what we found in Marmenegao Lagoon, we are measuring young of year sizes, which will have a disc width less than 2 meters. We also, if you look at the middle of the graph and the lines of maturity for male and female, the middle blue section shows that we have a lot of that juvenile turning into sub-adult range as well. So while the lagoon is used by young of year, it's also heavily used by these juveniles that are growing and getting older and using the area, so a potential secondary habitat. For the first criteria, we have to compare Marmenegao Lagoon against our other sites that I showed you in the first map. So I'm going to draw your attention to the bottom section, which is the inner lagoonal area, and you can see that in both the light blue section and the darker blue, the young of year and the juveniles are higher in Marmenegao Lagoon itself. So we believe we can tick off criteria 1. We then used the photo identification to calculate residency indexes. We did this on a seasonal basis. Like I said, they found they were more present during the northeast monsoon. And so we calculated this by looking at their sightings over a season and the total number of survey days over that season to get the graph that you see here. So the green color is the inner lagoon. We've compared this with the fringing area of the lagoon and then the rest of Ra Atoll as a comparison. And again, the young of year and the juveniles are exceedingly higher for Marmenegao Lagoon itself. So again, we believe we can tick off the second criteria. The third criteria asks us, is this area used over time, repeatedly? Again, we have all of our young of year IDs on the y-axis, and the green color shows that they're repeatedly seen across all survey years. The area has been designated as an ISRA, which is really exciting, but unfortunately it's still not a marine protected area in the Maldives. We are working on this. We're told it's going to be protected, but it's a lot of work going on behind the scenes. I know I'm short on time, so I just wanted to talk about our next steps, which is looking at where they go when they grow up, their ontogenetic changes, why Marmenegao Lagoon. So we're collaborating with oceanographers on why this area is so special. And one that might excite you is I'm supervising a student who is developing an AI, which is going to be open source to everyone, and it's looking at the boats and mantas interacting with each other and what distance and what speed we may have these negative behavior changes. That's everything. It's lovely to speak to you, and thank you very much.
Thank you very much, Jess. No time for questions again, I'm afraid. All right, so one speaker left in this first part of the session. I'd like to welcome Lola Riesco to the stage. She's got probably, or in my opinion, the best title for the session, One Model to Rule Them All, Discovering Mobulidae Across all the tropical Atlantic. Over to you.
Thank you. Hello everyone, my name is Lola Riesgo. I'm a PhD student in the Spanish Institute of Oceanography, and today I want to share with you why one model can rule them all. So, as we all know, the conservation status of Mobulis is not the best, but there has been in the last year an increase in international efforts to protect their populations. With agreements that do not only regulate trade, but also aim to protect and to reduce fishery impacts by binding their retentions and promoting safe release practice. So, although considerable efforts have been made to increase the scientific understanding that support the international agreements, there is a research gap, especially in the Eastern Tropical Atlantic Ocean. This is one of the most productive regions of all the Atlantic, and all the West African countries play a key role in the trade routes of Mobulid's products, and also is known as have a lot of hotspots of elasmobran biodiversity. So, all of this taken together highlights the urgent need to assess what is happening here with Mobulis, specifically with Mobulis occurrence. So we have to solve three fundamental questions: why, where, and when Mobulis occur. And to solve them, we are going to use the best tool that are species distribution models. So the research objectives here are study the spatiotemporal distribution of two Mobulid species, the Mobulata paracana, Mobula mobular, and the entire family. Then understand what are the key environmental drivers, and what is better to provide to the stakeholders, a species model or a family model. So we use Bycatch data from the European tuna purseine fleets collected through the European Data Collection Framework that operates in the high seas of the East Tropical Atlantic Ocean, and we match this data with the environmental data obtained with the Copernicus Marine data products. So to select the most relevant predictors, we applied a step-backward approach combined with different machine learning algorithms, and this allows us to obtain a final set of candidates for the models. So, at the end, both at the species and family level models show consistent responses to thermal fronts and to epipelagic micronecton concentrations. So we use a Bayesian spatiotemporal approach that are GAMS coupled with INLA that can deal with complex nonlinear relationship and handle spatiotemporal autocorrelation issues. So we have the fitting part to obtain the partial plots and the spatial effect, and we also have the prediction part that we are going to obtain the distribution. So to understand if it was better to use a family level or a species-specific model, we calculate the average of the distribution of both species and then compare it to the family through an overlap index that ranges from 0, that is no overlap, to 1, that is a complete overlap. So we are going to, I am going to show you 3 types of results. First, the partial plots that inform us about the environmental drivers, the distribution maps, high probability areas of occurrence, and then the overlap, we are going to choose what is better. So, results of the partial plot of the family level. I'm going to show you only the family level because we observe a very high overlap, and due to time constraints, I only can show you this. So, epipelagic micronecton, fronts, thermal fronts, chlorophyll show a positive relationship with Mobulis occurrence, and they are more likely to occur in moderate warm waters. Then the distribution maps. So we can observe a prominent hotspot in the Mauritanian coast, in the high seas areas offshore, and then in the Angola coast. Regarding the temporal part, we can see a persistent hotspot all year around in the Mauritanian coast and seasonal variation in Angola and the high seas, with different intensities between May and June and October to November. So the overlap, well, once we have mapped each of the species, we compute the average and then compare it to the family model level. And we obtain the overlap index that varies from 0.8 to 1, which means complete overlap. So what all of this means? The ecological message. We have seen that all of the environmental predictors are related with productivity and frontal dynamic features in the ocean. For example, in the Mauritanian coast, we have a lot of thermal fronts that boost primary productivity and increase the concentrations of zooplankton, and is the same for offshore areas. And in the Angola region, for example, we have the Angola Dome, that is a semi-permanent oceanographic feature that support a lot of local productivity. So they aggregate there to feed. So we think that this is the potential driver of this overlap. The tropical Atlantic Ocean is an oligotrophical environment, so there are only a few areas that have a lot of high productivity, so they tend to occur in the same areas to feed on the same prey. So why grouping? What are the management implications of grouping? So on board, observers doesn't have enough time to identify at the species level, so misidentification can occur. At the same time, in the case of our study, all of the species are in the ICAT convention areas and are aimed to the same regulations. So by grouping a species at the family level, we can simplify the information provided to the stakeholders. Also, this came with trade-off because we have increased the sample size, but at the same time, we have reduced the taxonomic resolution of it. This is not a universal approach. Taxonomic aggregation should not replace a species-specific assessment if you have reliable data, but in our case, it was our solution. So we are planning to expand the model for all the Atlantic and cover the whole distribution of mobulids. So I want to make an international call for collaboration. Anyone who has data, please came to us. We want every kind of data because we want to build an SDM for the entire Atlantic Ocean to understand where are the hotspots for this species. And if you want to know more about the ICAT policy context for mobulids, please came to see this poster. It's from one of my colleagues that is also called Lola, and I'm going to be there also if you want to chat with me about the data collaboration. Thank you so much.
Thank you, Lola. We do actually have time for a question, if anyone would like. There's a couple here in the middle. Sorry, Lola, you tried to get away with it.
Hi, great talk, loved it. I was just wondering, since you're looking at this as eventually translating into policy, what is the potential for using static SDMs and moving it to a dynamic oceanscape?
This was not an static SDM because we have modeled it was a spatio-temporal part, so we have like the picture of each month of where are all the mobulids, all the level mobulids, but so for me it's not an static SDM. Obviously all the models are static because it's like a shot of a specific moment in time, but if I have more data and I can translate it into the future, into understand what is going to happen maybe when climate change, how it's going to affect all these species. So, but translated into the management, well, now we have only static management measures. For example, banning retentions and safe release practice are static, they are not dynamic in time. So SDMs static and management statics are coupled. I don't know if I have answered. Oh, okay. Okay, thank you. I don't know. It's okay?
Yeah, thank you, Lola.
Okay, thank you.
Well done. Okay, so next up is Asia Armstrong. Asia's going to be talking about satellite telemetry revealing fine-scale movement ecology of reef manta rays in eastern Australia. Over to you.
Thanks so much, Guy, and thank you all for coming to listen. I've been slightly terrified about standing on this stage for 2 days now, but we're here and it's exciting, so thank you for coming, and a massive thank you to Daniel and all the team for this amazing conference. It's been incredible being here. I'm a first-time Sharks International person, so it's really cool. Okay, so today I'm going to be talking about some satellite tracking work that we've been doing on the east coast of Australia. These are really quite preliminary findings, so there's still a bit of work to do in this space, but you guys get the first preview. Nope, oh yep, here we go. Look, we're in the Manta Matters session. I'm not really going to introduce this species in any great detail, but what I will say is for the east coast of Australia, we are in a bit of a fortunate position where there's no targeted threats to this species, and it's assumed a fairly stable population, so it's really interesting for gathering baseline information on the species. So we've been doing that for a long time. Project Manta has been operating on the East Coast for about 2 decades, and we've done amazing long-term monitoring, looking at photo ID and citizen science input, and if you want to know more about that, you can come and see our poster in the next couple of days. And so with this work, we've really identified key aggregation sites and kind of those broad spatial overviews, but what we're lacking is really those localized movements and their fine-scale habitat use away from those key aggregation sites. So the aims of this study are to investigate their offshore movements, explore connectivity pathways between those aggregation sites, identify their fine-scale habitat use, and determine depth use and diving behaviours. So the main study site is Lady Elliot Island, which is in the southern Great Barrier Reef, and this site is known as their largest aggregation site on the East Coast. So it's a really important site because it's located in proximity to this indentation of the continental shelf. So we have this East Australian current, which is quite a nutrient-poor western boundary current flowing down the east coast. And then just offshore of Lady Elliot, we have this mesoscale feature, the Capricorn Eddy, where it actually upwells this productive water, and it's assumed that manta rays actually aggregate in this area to make these offshore movements into the Capricorn Eddy to access this productivity. So we put a bunch of tags on reef manta rays, deploying them with the modified Hawaiian spear on the dorsal surface, and I'm going to present some preliminary results from that. So the method, just so you know, we use these GPS fast lock splash tags, and so we deploy them with quite a long tether so that it floats above the animal, and so you're maximising those positions. And I just give you a nice example from this beautiful drone footage that somebody sent me of a tagged animal. So you can see when they're doing this surface lulling behaviour, that tag's actually breaking the surface quite a bit and giving us those beautiful positions. So over the course of about 5 years, we deployed 15 splash tags. Most of them were deployed at Lady Elliot Island. Just this summer, we deployed a couple at North Stradbroke Island, an aggregation site further south. And we tried to deploy them across all seasons and across males and females as well. We were super fortunate. The currents along this coastline work in our favour, and we were able to actually retrieve 10 of these tags. They washed up on beaches and people returned them to us. So we've got 10 archival data sets, which I'm going to be presenting the preliminary results from today. So this figure here, on the left-hand side, you can see those GPS fixes, and then the right-hand figure shows the positions that are interpolated using the GPE3 modelling and their depth use as well to give you a look at those tracks. So the average tag duration was about 50 days. Some tags stayed on for up to 124 days, the full deployment. And the animals travelled up to 700 kilometres during this time. What we found quite interesting is that they actually stayed relatively close to their tagging location, regardless of season, and they really made limited offshore movements. So we wanted to have a little look at what their depth use was like during the tagging period. And so you can see here their vertical movements. So this is for the 10 individuals. Most of them stayed within the top 50 metres of the water column. There are a couple of individuals that did make some deeper dives down to 300 metres, and so those ones were moving off the shelf. But the others, most of them stayed within that top 50 metres with an average diving depth of about 20 metres. So then I wanted to have a look at what these dives were like. Are they foraging with these on-shelf movements? So I basically classified the dives to if they were over 20 metres, because that's kind of accessing that mixed layer depth in this region, and looked at U-shaped diving shapes for foraging or V-shaped for transiting. And this is what that looked like. So you can see there is a bit of variation between the individuals, but as you can see, a lot of that U-shaped diving is still happening in those animals that are only diving to depths of up to 50 metres. So they are foraging in those shallow waters. And then I wanted to see if there was a dial pattern to those dives. And so you can see here that the night is shaded on the figures, and the mantas really don't exhibit a clear pattern. There's a lot of variation between individuals. So as a lot of you in this room would know, manta rays feed on zooplankton. Zooplankton undergoes the largest synchronised vertical migration every single night. So we generally assume manta rays do reverse dial migration, going down to feed on the zooplankton at night, and we did see that that was the case in some of the animals. This is an example of the animal that did those deeper dives, but there was a real mix. Some of them were diving during the daytime and some were doing a combination of the two. So just to show you a summary of that, you can see that there's quite a even split between daytime and nighttime diving behaviour for this species. So just to summarise, we were really surprised by the limited offshore movements here and the shallow diving behaviour without any strong dial signature. But I think that it's an example of really taking in your local context when you're looking at these results. So we assumed they were making offshore movements to the Capricorn eddy, but instead we think the Capricorn eddy is likely bringing those nutrients from depth and pushing them up onto the shelf, where we're seeing the manta rays foraging to the south of Lady Elliot Island. And so, yeah, this is just preliminary. We want to look at drivers for these movements and, yeah, explore chlorophyll A concentrations and associated with the movements and a range of other things that didn't happen before the conference. So with that, I would just like to thank all of the supporters and funders for this work. I'd like to do a special shout out to Mark Erdman, who has been a wonderful supporter of this work, and to Lady Elliot Island Eco Resort for their incredible partnership with Project Manta. And thank you for listening.
Thank you very much, Asia. We have time for one question, if anyone would like to ask one. Jessica, I see, has her hand up. Has anyone got a microphone? Thank you.
Great talk, Asia. I was curious about your placement of the anchor of the spot tags and or the splash tags. Is that so it reaches the surface easier? And I didn't know if you had any comparisons of where you place the tag on the animal's body.
Yeah, so we've done quite a bit of tagging historically with acoustic tags and mini-pat tags, and those ones we don't have a very long tether on them, and you generally position them a bit further down on the animal's body. But through a lot of experimentation before me doing this, Mark can attest to this, if you put one of these tags further down on the body and you have that floating tether behind the animal, it pretty much gets hammered immediately and removed from the animal. It's like a fad and predators just go for it. So putting it right up there on like the shoulder kind of musculature, it allows it to not look so much like a bit of prey floating above that animal. Yeah.
Thank you very much, Asia.
Awesome, thank you.
Okay. Next up, we have Simon Hilborn. Simon's going to be talking about oceanic manta rays passing through paradise in the Maldives.
Perfect. Good morning, everyone. Yeah, my name's Simon, and I'll be talking about oceanic manta rays in the Maldives. So, as we've covered today, oceanic manta rays have a conservative life history traits. They are long-lived, they are late to reach sexual maturity, they only have one pup per pregnancy, and those pregnancies are often have extended periods in between them. As a result of this and pressure from fisheries all around the world, bycatch and targeted fisheries, oceanic manta rays are now listed as endangered on the IUCN Red List. Oceanic mantas are widely distributed throughout the Indian Ocean. They're found from South Africa, up the coast of East Africa, into the Arabian Sea, and down the coast of Thailand, Indonesia, Western Australia. In the Maldives, which hosts the largest population of reef manta rays in the world, oceanic manta rays have been known for a while, but until recently, they've been relatively understudied. The Maldives is also a protected area for manta rays, which is great, but unfortunately that protection doesn't extend beyond the EEZ of the Maldives and into the high seas and into neighboring countries. There are risks from fisheries across the region. This project is looking at the movements and demographics of the oceanic manta population in the Maldives, or the one that's been documented in the Maldives, and it uses a long-term photo ID data set. All of the data presented here is based on where we have photo ID data of the unique spot pattern, so we aren't including IDs for which it was just a photo of the dorsal side or a report of the individual. It's only with confirmed ventral IDs. In addition, we also managed to deploy 3 mini-pat tags last year in 2025, for 120 to 180 days, so I'll present a little bit of that data too. So from the photo ID database, we've recorded over 1,100 individuals in the Maldives from 1,250 or so sightings, which makes it the third largest photo ID database for oceanic manta rays in the world, after the Ecuador-Peru and then the Revillagigedo archipelago databases. So it's a pretty significant population. It slightly has a male sex bias ratio, and it's predominantly adults making up the population. We've recorded a handful of pregnancies over the years, but not really that many, and about 2% of the population is black morph or melanistic. In 2010 and 2015, the Manta Trust had some research expeditions and got opportunistic sightings of aggregations of oceanic manta rays in the south of the Maldives. But it wasn't until 2018 that we started a kind of dedicated project looking at this species in this region. Shortly after that, in 2019, we had our largest season. We recorded over 300 individuals in a 4-week season with up to 18 photo IDs on a single 40-minute scuba dive. So it was pretty nuts. There is seasonality in the sightings across the Maldives. We have recorded mantas, oceanic mantas, throughout the year, but the sightings start to pick up around December through to January, reaching a peak in March, April. So there's definitely a hotspot of sightings activity around that period. When we look at the behavior of the mantas, when we encounter them, we have recorded feeding behavior, we have recorded cleaning behavior, we have recorded a little bit of courtship, but by and large, all of the sightings, about 90%, the animal is just cruising. It's just swimming around in the surface waters, in the deep, off in the reef, over the top shallow reef, kind of anywhere and everywhere. In terms of distribution of sightings, the Maldives is made up of 26 geographical atolls, and we've now recorded oceanic manta rays from 18 of those atolls. But there is a massive disproportion as to where those sightings come from. Over 80% of the sightings come from Formula Atoll in the south of the Maldives, which, as many of you will know, is just a single island. It's an atoll by itself, but it doesn't have the lagoon system. So it's definitely a very special place for this species. We've recorded a number of re-sightings. About 90%, over 90% of the individuals have only ever been recorded once. 8% have been recorded 2 or more times, so we have re-sightings of these individuals. And from the graph on the side, that's just the manta's ID. But most of the re-sightings are over a short period of time, a couple of days, maybe a week, in the same location. We have, however, recorded one individual over an 18-year period, which is pretty awesome. And that individual has been recorded 4 times now. And that's also the most sightings of a single individual is 4. Of the re-sightings, we've got about 60 re-sightings. And of those, about 20 of them have resulted in what we call an inter-atoll movement. So the initial sighting was in one atoll, and the subsequent sighting was in another atoll. And as you'll see, most of those re-sightings happen within the central north atolls, with a little bit of connectivity to the southern region. So with this kind of seasonal appearance of oceanic manta rays around Formula in a seasonal fashion coming back every year, we deployed the satellite tags in the hope that we would see this kind of seasonal movement, all these mantas using a common kind of migratory route and returning every year. The first satellite tag that we deployed traveled over 1,600 kilometers out towards Sri Lanka. It did, well, depending on the uncertainty, it possibly crept into Sri Lankan waters, but it definitely left the Maldives into the high seas. Unfortunately, the second manta then decided to do something completely different and went west instead. And the third manta, which I should caveat was a juvenile, decided to do something different and stayed central to the Maldives. So although we didn't document any commonality in the seasonal movements or long-range movements of these individuals, what we did record was the fact that they are traveling very long distances, up to 1,600 kilometers. The movements vary greatly. We only have 3 examples, but all 3 did something completely different. And they spend varying amounts of time within the protection of the Maldives EEZ. Between 7 and 100% of their tracks were in the safety of the water. And we also documented potential international movements from the Maldives over to Sri Lankan waters. Why is this concern though? Again, I come back to the beginning of the talk. Sri Lanka hosts the largest Mobulid fishery in the world. We've documented over 1,000 individuals over 9-10 years of photo ID data. But that same number of individuals, around 1,000, are caught and killed every single year here in Sri Lanka. So it kind of highlights the urgency of this work. So just to wrap up quickly, the Maldives hosts a globally significant population of oceanic manta rays, with Formula Atoll in the south of the country being a particular hotspot for these individuals. Low residency, low resighting rate, long-range movements, and behavior all suggest that these animals are transiting through at least the shallow waters of the Maldives where scuba diving activities are happening. And these mantas are likely, or they are moving into areas beyond national jurisdiction where foreign fisheries are likely targeting these individuals and landing them. I'd just like to say a big thank you to some of the funders, and if you have any questions, please let me know.
Thank you very much, Simon. And we do have time for a couple of questions, so if anybody would like to ask Simon a question, now is your chance. One at the back.
Yes, thank you, Simon. Why is Fuvumula such a hotspot?
That's a good question. We're not entirely sure for now. The atoll is quite unique to the rest of the Maldives. It's a bit of a pelagic magnet. There's a bunch of presentations, talks, and posters at this conference. It's a hotspot for thresher sharks, tiger sharks, hammerheads, all sorts of kind of larger pelagic animals. So it's perhaps the kind of pelagic nature of this island. It's a small island surrounded by deep water that attracts these large megafauna to the area. Why the manta rays are turning up there, we don't really know, because they're not feeding there, they're not really cleaning there very much. There's a little bit of courtship, but it doesn't seem to be the primary driver. And if it was for any of those reasons, we would expect the same mantas to hang around for a little while, and we'd see the same ones day after day for a period. But that's not the case. The ones we see today, we go out diving tomorrow, they're not there, but it's completely different mantas, and they seem to be transiting through on some sort of migratory pathway. We put the tags out hoping to figure out what that was, and it came back with 3 completely different tracks. So we're still a little stumped, but more work to do.
Thank you.
Thank you very much, Simon. Okay, bear with me. Next up, I am presenting. Tonti, who was going to do this presentation, was unable to make it, and the work I'm presenting is actually primarily undertaken by Hannah Maloney, and Hannah's just had a little baby boy, so she couldn't be here. So you have to bear with me because I haven't had time to really go through the presentation, and I'm not that familiar with it. So excuses out of the way, I'll do my best. Just need to start the clock. So the aim of this study was to repeat and improve upon work that I undertook with Chaz Anderson and a few others over a decade ago, really to try to estimate the economic value of swim with tourism in the Maldives. There was also a global study, I mentioned it in my presentation earlier today, that estimated back in 2012-13 the global value of manta ray diving. Primarily that paper produced results of $140 million US a year were being generated through these swim with activities. And the Maldives study that I undertook with Charles was about 15 million at the time for that country. So we wanted to repeat this process. Tourism has massively increased in the country over that time, and we wanted to sort of use this evidence to push the government to implement better regulations and management around around these activities, and to try to demonstrate globally the importance and the significance of this economic income. And just to sort of, just to highlight that some of these, some of this work also feeds into some of the UN's development goals. So the first thing we wanted to do was
assess the actual revenue, the economic benefits. We wanted to also describe the distribution of this, so where is it actually focused in the country, and then discuss the sort of the social output and values of this. So we used data from the Maldives Ministry of Tourism. We also tried to work across the country, and we undertook a large number of surveys with operators, and we also used the internet extensively and all of the expertise within the organization to try to gather as much information as we can that was freely available. For example, how much a specific dive, a manta dive or a snorkel was advertised for, and then all of the surveys we were asking information on, you know, the amount of times they went out each day, each week, etc., the number of guests they took, the amount they were paying for the different activities. So a huge amount of effort by gathering this information, tracking down the number of different hotels and the different resorts. We grouped them into 4 main types. There's a sort of the standard resort, the luxury high-end resorts, which charge an awful lot more for their activities. Then there were the local islands, which have sort of dive centers and operations from those locations, as well as the liverboards. And the liverboard diving industry is huge in the Maldives. And you can see the numbers there. There's a large amount of surveys and a large amount of effort that went into collating this information. And we also grouped the activities into snorkeling versus diving. There was different sort of outputs and different amounts charged for these activities as well. So we did all the calculations, and this is certainly an area that I'm not that familiar with, but we put together all of the different costs and expenses associated with these activities to come up with a total figure. We'll talk about more about that in a second. And we also asked questions about the amount of marketing and the amount of significance that these operators put into these activities. And as you can see, there's a lot of the resorts focus heavily on branding and marketing around the manta rays. It's a big appeal to a lot of the people that come to the country that want to get in the ocean, to want to see these animals. And so we're starting to see increasingly, you know, sculptures, resorts being shaped around, named around, promoted for these very charismatic creatures. So 80% plus of operators offer diving and snorkeling with these animals, and 80+% of them also market these animals significantly. Here's the distribution of the activities, and I would note here that this is reflective of where tourism is most developed in the country, but more significantly than that, it's a reflection of where the mantas are present in the greater numbers. I talked earlier about Hanif Fadzli, which is in Bar Atoll, so you can see down at the bottom there's a big spike in the amount there of operators and focus on these activities. So it's the two are sort of reflecting each other. But as you've just heard from Simon's presentation, this distribution is also segregated by the different species as well. So there's a different focus to formula where the oceanics are compared to the reef mantas that are further centrally and northerly situated in more abundance in the country. So this affects the activities that are undertaken by these operators throughout the country. So we tried to reflect all of this information as well. And here's just one case study. Again, I talked about Hanif Fadzli earlier and how significant this site is. The stats down on the bottom right show that on average trip, so just one trip per operator, they're taking around 53 tourists and they're sighting 32 plus individual rays during that excursion. So there's a really high pressure of tourism activity at this site, but it's obviously because it's such a special location to see so reliably and predictably these animals. And that's resulted in management around this site. So an MPA has active management with rangers on site. There's a fee to access the location as well. So you've got to pay for your tourist operation, but there's also a fee that enables the management to be taken, to take place at this site. Okay, so the figures, the combined economic revenue for the country has gone from 15 odd million to 227 million in the last decade. So there's a huge increase in the revenue being generated. Tourism is just massively expanded during the time that I've been working in the Maldives. Here we go, you can see that massive increase. And so the next study we want to do is update Mary's 2013 study globally. So we'll be applying these methods that we've redone in the Maldives, and we're already working on other countries around the world. And we want to try and do a re-snapshot of the current Manta, Devil Ray, tourism economics globally. And I can certainly anticipate that figure on the right, 140 million times by 10 or so as well. So these animals are certainly of significant value to the economies of many of these countries around the world. Okay, so this just highlights the proportion of the share of contribution in terms of tourism and GDP overall is stayed around the same, but you can see that the mantas as a ratio of that has increased significantly. And then finally, we want to talk about how this is important for the Maldivian communities. There's a large proportion of Maldivians obviously are employed in the workforce, in the resorts, in the diving and snorkeling industry, and staff salaries alone are generating 27 million is being generated from these manta dive and snorkel activities. And culturally, these animals are also really, really important. It's driving protected areas, biosphere reserves around these charismatic species, and it's a species that is of significance to the communities and the country. Some more additional maths we did to take into consideration other total economic benefits, pushes that number up even higher. And also speaking to the respondents of the surveys and the communities, and the tourism industry does exit surveys of tourists when they're leaving the country to gather information on what was the most important must-see reason for their travel. And repeatedly, when you ask the divers, you ask the tourists leaving the country what they wanted to see most, what they valued most in terms of the marine life, manta rays were right up there at the top. We see that most of the resorts, a lot of them have marine biologists, and those marine biologists are giving presentations on these animals. And the local communities value these animals. They're a species that is increasingly seen as a symbol of their cultural heritage. So takeaway, mantas are worth a lot in the Maldives. That's no surprise, but the amount is increased and has become a greater share of the overall GDP. And obviously, this is important for helping us drive conservation measures, because money talks, and that's obviously an important aspect of the work that we do. So, thank you very much. Thank you, Hannah, for putting this together.
I don't think there's any. Does anyone have any questions? I don't think I have time, but. Yes, no. Who's next? All right, Mel, you're up. So Mel Cronin, she's going to be talking about how do we turn fisher innovation into conservation impacts.
Okay, hi everyone. My name is Mel Cronin. I'm a new faculty member at University of Massachusetts in the Northeast US. And I'm super excited to be here to tell you all about some work we've been doing among some collaborators, identifying and trying to scale innovations for Mobulid bycatch mitigation in tuna fisheries. And I want to begin by reintroducing you to the Mobulids, which you know very well by now, but the manta and devil rays, which are characterized by extreme charisma, but also slow growth, low fecundity, and unfortunately global population declines. By the way, that's Marta, our keynote speaker from this morning. And one sort of understudied source of Mobulid mortality is from the global tuna purse seine fishery, which is the source of roughly 70% of the world's tuna, mostly for canned, shelf-stable tuna products. We estimate that roughly 13,000 individuals are caught annually in this fishery, although there's not a whole lot of great estimates. And in the Eastern Tropical Pacific, which is a Mobulid biodiversity hotspot, we know that bycatch in these fisheries occurs from the coastal area well out into the pelagic areas. Each dot on this map is a Mobulid bycatch event. And 5 species are caught, all of which have significant vulnerability. And I want to show you sort of what this looks like when one of these animals is caught on board. So you see here, this is a Mobulid bycatch that occurred at the very beginning of this project before we had done any sort of intervening. And this is an example of a really bad release, and I'll tell you why. So you can see everyone's trying to grab this animal. There are like 7 fishers trying to grab this animal, which is extremely slippery. There's really nowhere to grab onto it. So as a result, they often grab onto its cephalic lobes, which are very sensitive. Sometimes they grab onto its eye, and then they're going to use a stretcher to pick it up and release it overboard, which can cause bending of the pectoral fins. You might have also seen earlier that the Mobulid actually knocked off the hard hat of one of the crew members, which is actually very, very dangerous on these fishing boats. Eventually it does get out to sea, but this is a release that we weren't really super happy to see. A couple other pieces of information or context were occurring right when we started this project that made it clear that we really needed ground up sort of grassroots bycatch mitigation and intervention. We knew that the existing bycatch policy was not sufficient, at least currently, to reduce mortality of these species. At the same time, fortunate for us, the industry was interested in an eco-certification and had been told specifically by Monterey Bay Seafood Watch that Mobulids were a problem for them, which was great for us. There was also some really great tagging work by a colleague of ours, Josh Stewart, showing that mortality is most likely to occur if the animal sits on deck after 3 minutes. So we had this 3-minute golden window that we knew we had to get these animals off. But as you saw, these are like a giant writhing bar of soap. So what could we do? Well, first we asked fishers. So we did a big survey effort with 170 stakeholders from the fleet, including skippers, crew, mechanics, anybody who knew about the operations. We led focus groups and workshops to elicit rapid release. We also gave fishers matching t-shirts with the species ID on them so that they could practice it when they look at each other. And the first thing we wanted to know was, like, what's the problem? Why can't you release these animals? They mostly said that those animals that were too heavy or large were really hard to release, and that they really didn't have the right type of gear to release these big animals. So for me as a Mobulid scientist, I wasn't really surprised by this because we know that the giant oceanic manta ray, the largest species, can weigh roughly the same as a Honda Civic. So that made sense. But we also found that in absence of the proper gear to release these animals, that they were using some harmful methods to release them, including putting a gaff into a gill slit and using a cargo net, which is slightly better, but as you can see in this video, this can cause bending of the pectoral fins, lacerations, and potentially damage. So we wanted to think about these large animals and how we could release them. At the same time, something really cool that came out of these workshops and similar workshops that were led by my colleague Jefferson Marua in the Atlantic were these really great innovations and the precursors to what we now call the Mobulid sorting grid or the Manta sorting grid. The really cool thing about this is that it acts like a pasta strainer, where the Mobulid is like the pasta or colander, and the tuna are like the water that go through. So the tuna are allowed to go through the slat, while the Mobulid is caught and can lie flat without bending its pectoral fins. So we were really excited to see this work, and so the next step we had was to test it. So along with colleagues, including Jefferson and Gala Moreno at ISSF, we distributed plans to construct the grid on 12 tuna purseine vessels in the entire US fleet. We also allowed fishers to use it when they wanted, but we didn't require them to use it every single time to kind of mimic a real life situation. And then we measured things like duration on deck, which we can include as a proxy for mortality. We did tag animals, but that didn't really work out, so we could talk about that later. And I want to show you what this looks like.
Yeah, but you guys,
sorry. You can see the animal is lying flat, and it's taking a little bit of an elevator ride to get down closer to the water, after which it will be released from the side of the grid. I'm here to tell you that this is warp speed on a tuna fishing vessel. So we were really pleased to see fishers actually using the grid. So how did it work? We wanted to see which animals were preferentially used by fishers with the grid, and we found that, no surprise, animals that were much larger, which are those on the left in purple here, were those that were released with the grid, while smaller animals were released manually. If it's small enough to just throw off the side of the deck, makes sense. But we also found that there was no difference in the duration of capture, so the time that the animal was on deck, between these two methods, using the grid and just a manual release, which is great because we can tell the fishers that this intervention is not something that's going to increase time of fishing, which is really, really critical. And even more so, we found that many of the grid releases were under that 3-minute mark, so this is a really promising finding for us. We definitely need more sample sizes, and fortunately some of this work is being tested using satellite tags, but this was a promising first finding. So what next? We're currently scaling the grid. It's now being used on at least 55 vessels in 3 oceans, and hopefully more soon. It was included recently in an amendment to proposals for this and other sort of best practices handling guidelines in 2 regional fisheries management organizations, so we're excited about that. And then we've also distributed these handy posters to many of the vessels, which are available for download at our website if you want some nice wall art. And I just wanted to very briefly reflect on reasons why I think this was a successful collaboration. I already told you that we identified and aligned this incentive in the eco-certification and that we harness existing innovations. Also, this is a really low cost and very, very simple intervention. These grids can be fabricated for less than $2,000 US, which is, for this kind of a fishery, really feasible, and they can be made with existing materials that are already on the vessel. Personally, I learned a couple interesting things from this work, which was certainly the importance of time at sea. These vessels go out for 2 months at a time, and those trips may not actually encounter a Mobulid, which was the case for one of mine, unfortunately. But it was really important for me to spend time to understand the fishery. I also learned to be a team player. This is a photo in the center of me getting my head shaved as a way to help the crew increase their fishing luck. And though that was a sacrifice, it really helped me learn about, getting on board with what the crew is doing. And then finally, because I was the only woman, and in fact, I don't think that there had ever been a woman in this fleet before, it was really important for me to ensure safe field access. Fortunately for me, I run an organization called Field Futures, and we provide training specifically to reduce the risk of gender-based harassment in field work. So please let me know if you're interested in that work. We do trainings all over the world, and I have stickers. Finally, I want to point out a couple student projects that are here, including one led by Izzy Osiander, who used genetics to document Mobulid meat trade, and then one by my student Isabella Garfield on individual decision-making and how it affects Mobulid bycatch. With that, I thank you so much for listening, and happy to answer any questions if you have them. Thanks.
Thank you, Mel. Love the Manta grid. Time for one question. Nope, okay, you're off the hook. Next up, we have Egin. Go Egin. Egin Salim, welcome to the stage. Egin's going to be talking about reducing Mobulid bycatch through community-based approaches in Indonesia.
Okay, so hello everyone. Thank you for coming, and it's a pleasure to be here to share with you about our work in Indonesia at Shark International. Yeah, finally, Shark International. So today we're talking about regarding how we try to reduce bycatch of Mobulids in Indonesia using community-based approaches. And to do this, I will take all of you to our primary location. It's called Muncar in Banyuwangi area. So it's very close to Bali Island. Mostly of you maybe know the Bali Island, the famous island in Indonesia. Is separated by Bali Strait. Bali Strait become the most dense area in this Lesser Sunda with the small scale and the industrial scale fisheries. So this place is quite unique because the highly fishing pressure and also mixed fishing gear there. And nationally, Bali Strait known as a sardine fishery. But on the other hand, this area also become a elasmobrang magnet. Why? Because a lot of coastal community here, depends to the elasmobrang meat they consume, and they trade, and they got elasmobrang for everywhere. So it's like a central of elasmobrang trade in East Java. And based on our survey, we recorded 66 shark species and 52 ray species. And a vessel boat is targeted shark and rays, but most of them is from the bycatch. So to get deeper with the bycatch, our team conducted daily landing monitoring for almost 6 years, from 2019 to 2025. And this is the result. As you can see, we have some peak Mobulid landing on 2021, and it's declining every year to 2025. We identified 5 Mobulid species, including Mobula mobular, Mobula trustoni, Mobula tarapacana. Still, there's Mobula birosis and Mobula arfredi. With the 3, the first one is the dominating. They all catch as a bycatch by drift gillnet mainly, purseine, and bottom gills. So, in addition to the biological data, we recorded 56% of male and 72% of female was captured with the immature condition. And by this data, we know also the peak season of the bycatch occur in August to October each year. And we want to know deeper about this bycatch. What is the main driving factor that causing this kind of bycatch in this area? So we combine this landing data with the socio-economic survey by using social ecological framework, and we found this super interesting one. So the bycatch number correlates with the fishing activity and also market price. So there are 2 main driving factors. First is market, and the second one is behavior itself. So we also asked the fishers regarding what if you releasing back the Mobulids to the ocean when you got it still alive, and they say, oh, it's impossible. The fish will be dead or it won't survive. So we test it actually. So we deploy 26 survivorship tags and found that the post-release survival rate is 86%. This evidence become our new hope that if we treat the Mobulids with the carefully and with the time-specific frame, it will increase the survivorship. And then yet we're realizing that the fisher itself become a key actor on this initiative, so we try to getting close with them. It's an easy work, so we deploy some steps to get more closer with the fisher community. We try, the first one is we try to get the better connection with their wives. It's a very strategic approach that we use in the field. And then after we getting close with their wife, we can go with the community development with the fisher itself. And these 2 approaches combine into one the solution that we try so far. It's called Sustainable Seafood Network. So why their wives? In Muncar, wives have a very special place in the fishers, on the fishing activity. Wife is like a manager. So actually the fishers just like, they manage all the things about the household, the fishing activity, even the day that they want to go fish. So sometimes the fishers like, oh no, I'm not going anywhere. And the wife said, hey, you have a debt, go to fishing. And then, okay, I will go. So it's very clear that we try to engage with the fishers first, fisher wife first. Then after that, the fisher's wife like, hey, you have to attend this kind of like NGO because they're so kind of us. They give me like financial literation workshop and etc. And then come up with the fishers gathering up. We built some group mentoring And from the group mentoring, they provide valuable insight and solution that emerge organically, solution from there. And it's called Sustainable Seafood Network. So we connecting them to the potential market in Bali because we are close to Bali. So the result is 96 activists signing MOU with us, doing some activity to reduce the mobulids. And we got 30 plus partners in Bali. Shout out to Bepasi restaurant, the first restaurant that want to support us, this one. And the evidence, it's quite interesting. From 96 activists on the previous year, before we do this initiative, they landed around 126 mobulids. And a year after, from this group, they only landed about 19 mobulids. So like 85% bycatch reduction. And this one, it's a video footage that, so we give them action cam to record all the fishing activity, and whenever they got the bycatch, they want to release them back to the ocean. Yeah, it's a little bit different with Mel's situation. It's like video from 1995. Yeah, basically, mostly they work alone, so no one helped them in the small boat. But every time they got the mobulids, we try, please getting back to the ocean as soon as possible. But he just like measuring them. So after the trip, he just like, oh, I got 5, what's it called? Yeah, 5 fingers times. And we also trying to identify the sex actually. And we try to give them some education. Don't use a hook or don't put your hand on the gills. And here the result. He carry it like a baby. So it's not a good way, but still we. So yeah, I will thanks to our previous project leader, Betty Lagebauer, and all the alumni and the funders that make it work happen. Okay, thank you so much.
Thank you again. Time for one quick question. All right, off the hook. Thanks again. Next up, we have Teen Boggio-Pasqua. Apologies if I've pronounced that. That was good. Okay, great. She's going to be talking about from the beaches to canyons, an integrative approach to the ecology of the Atlantic pygmy devore to support ISRA proposals in the Gulf of Mexico.
You guys. Hi, everyone. Thank you so much for being here. I'm very excited to share some of my PhD findings in front of such a large aggregation of Mobula enthusiasts. As you can see, this work is the result of a very collaborative effort. And before I begin, I'd like to acknowledge my amazing co-PI and mentor, Kim Bassos-Hall, who has been paving the way for this research at Mote Marine Lab for over 10 years now. So yeah, let's dive in. You probably guessed it from the name. Mobula hypostoma is one of the smallest mobulid rays in the world, but also one of the least studied. These endangered rays have a wide neretic distribution across the Atlantic, yet they're not so easy to find. And despite their distinctive small size, they have been actually continuously confused with other mobulids and misidentified in bycatch records. So as a result, very little is known about their biology and ecology specifically, including population status and most critical habitats. My PhD aims to fill as many of these gaps as possible, including supporting targeted conservation at multiple scales. To do so, Kim and I partnered with Okaloosa County, Florida researchers in the northern Gulf of Mexico, because in this area, Mobula hypostoma have been continuously been recorded in the past few years. And this is what it looks like. So groups of them, not as big as Munkiana, but still chain feeding in the surf zone, and yeah, it's pretty spectacular. So this unique aggregation has allowed us to develop a dedicated project since 2022, and among the many aspects that we were able to study, the following questions are particularly relevant for ISRA designation. Is this aggregation seasonal and predictable? What prey items are they feeding on, and is prey availability driving their presence? Which life stages use this aggregation area, and where do they come from, and where do they go afterwards? To answer these questions, we adopted an integrative approach. We compiled social media and citizen science observations from the area, and we conducted targeted fieldwork, mostly in the fall, when devil rays seemed more abundant. To catch rays, we encircle them with a 50-meter diameter seine net and send a snorkeler in to gently scoop them up. The rays stay submerged 99% of the time, thanks to an onboard tank and a floating net to temporarily hold the excess individuals. Each ray is measured, weighed, pit tagged for re-identification. We collect a range of samples, including cloacal swabs for DNA, fecal DNA metabolic coding, but also fin clips for genetics, muscle, blood for stable isotope analysis. And finally, we deploy two types of tags, passive acoustic transmitters that we surgically implanted, and PSATs, pop-up satellite archival tags that we attached externally using umbrella darts. Since 2019, we have tagged 74 devil rays, mostly juveniles and subadults, within the aggregation area. Thanks to the iTag and FACT networks, which facilitate data sharing among array and tag owners in the region, we now have over 100,000 detections from 59 devil rays, which is really exciting. 3 major detection hotspots emerge from this data set: the Northwest Florida aggregation, as expected, but also the Mississippi Barrier Islands to the west and Sarasota, Tampa Bay area to the southeast. Within the aggregation area, we noticed a clear seasonal signal in the proportion of available tags detected, with a major peak in the fall and a smaller peak in the spring. And we found pretty much the same pattern in the hundreds of observations compiled from social media and citizen science platforms since 2011. We then calculated a monthly residency index defined as the average proportion of monitored days each ray was detected at a given month, and we found that juveniles were more resident in the area than subadults, but for both, residency followed the same seasonal pattern, highest during the fall and followed by the spring. Now, what about the adults? Well, we rarely encountered them. Unfortunately, we only could tag 3, and those individuals were rarely detected anywhere, suggesting a limited use of shallow coastal waters, where most receivers are deployed. When grouping receiver by distance to shore, we also noticed a clear dial pattern in coastal habitat use, with rays being mostly detected inshore during the day and moving offshore at night. Now, inshore, we identified there you go. We identified dense patches of mysid shrimp, Metamysidopsis swifti, which is typically found in high-energy coastal waters and has a peak reproductive period in the fall. So DNA from this mysid was found in 80% of the rays we sampled using cloacal swabs, but we also found some fish DNA in some of the rays from coastal species, and 2 of them are known to spawn in the fall, which means that rays probably also feed on fish, eggs, and larvae. Now, what are these rays up to when they move offshore? We deployed 14 PSATs, mostly on subadults. Unfortunately, we had some premature tag detachment issues, which led to shorter deployments, but we still were able to obtain valuable insights into their vertical and horizontal habitat use. Overall, most of the rays remained on the continental shelf and used the upper 50 meters of the water column, but some of the individuals performed deeper dives down to almost 200 meters maximum, and all of these dives occurred at the head of the DeSoto Canyon, which is located approximately 30 kilometers offshore. Here's a quick example, a subadult male that spent a few days there. You can see a clear dial pattern with greater vertical movement during the night, which might reflect foraging on dial vertical migrations of zooplankton, while the daytime depth is more stable and might be associated with a more stable layer of prey at depth during daytime. Now back to the acoustic telemetry data. If we zoom out and consider all detections, this is what we got for every single tagged individual. So here detections are plotted over time and color-coded by region to help visualize movements. And you can see that juveniles were mostly detected in zone 5, where the aggregation area is, while subadults and adults were more wide-ranging. Most notably, 9 subadults and adults performed long-distance seasonal movements between the 3 major hotspots I showed you earlier. This is our most striking example to date, a subadult male we tagged in Sarasota in October 2021, which repeated these long-distance movements over several years. And these are pretty fast. The 500-kilometer journey between Sarasota and Northwest Florida can take as little as 8 days. Southward movements towards Sarasota typically occur in the summer to early fall, while the northward movements typically occur from mid-fall to early winter. Now, interestingly, we haven't received any detections from the Florida Big Bend region, which is that northeast corner, which means that individuals most likely follow the shelf edge instead of the coast. Now, back to our initial goal, ISRAS. This work led to 2 proposals. The first focusing on the acoustic telemetry data to delineate a migration corridor connecting the 3 hotspots. And I'm very excited to share that just last week it was accepted by the ISRA independent review panel, so yay. Thank you. The second proposal described the feeding aggregation area in Northwest Florida, and it's currently under review, so we don't know, but we're very optimistic given the multiple lines of evidence we have. So if accepted, these 2 ISRAs are the first identified for the species worldwide, which is really exciting, but there are many more knowledge gaps that remain, both in our study region, but also across the species range. In the northern Gulf of Mexico, future work could focus on confirming additional feeding grounds, for example, and probably the 2 other hotspots that we identified, but more importantly, mating and pupping grounds are largely or completely unknown in the region. We did not detect any neonate during our fieldwork, and that's over 130 individuals examined. From a conservation perspective now, we aim to help integrate this ISRAS into marine spatial planning, and we have already begun doing some outreach and raising awareness within the aggregation area, because despite state-level protection in Florida for the species, we know bycatch by anglers still occur, and in sometimes illegal retention and use as shark bait. Now, this raises the question, beyond Florida waters, the species is not protected, so we might wonder if it's worth applying for a federal protection, like manta rays, for example, which have been listed under the Endangered Species Act since 2018. And that's it for me. Thank you so much for your attention. I'd just like to thank the funders, Save Our Seas Foundation, Mozarteam Foundation, and the 2 aquariums listed here, and all the many, many, many fantastic collaborators we work with. This really takes a village, and Marta and Chris for their help with the ISRA proposals. Thank you.
Thank you, Tine. No time for questions, I'm afraid. Now, unfortunately, Mohammed Abudaya could not be here, but we do have his video via recording. So hopefully the moderators can get that up on the screen and start it. Mohammed's going to be talking about insights into the spine-tailed devil ray spatial ecology in the Mediterranean through satellite telemetry. Do we have the video?
Good morning, everyone. My name is Mohammed Abudaya, and I am pleased to be with you on behalf of the authors of our published study. This presentation summarizes recent evidence on the spatial ecology of the devil ray, Mobula, in the Mediterranean Sea. The study uses satellite telemetry from 9 tagged individuals to examine seasonal movement, habitat associations, and conservation implications. The central message is that this endangered species appears to make predictable basin-scale seasonal movements, concentrating in the Levantine Sea during late winter and early spring, while using the central and western Mediterranean, more broadly during warmer months. The aim of the study was to investigate whether spintail devil rays in the Mediterranean show seasonal migratory behavior. To do that, the authors tracked 9 individuals tagged between 2016 and 2021, with animals tagged off Gaza and in the Strait of Messina. They then combined movement tracks with kernel density estimation and generalized additive models to relate occurrence probability to environmental variables, including sea surface temperature, chlorophyll, salinity, and zooplankton. This slide summarizes the deployment dataset. A total of 9 rays were tagged. Tag durations ranged from 28 to 330 days, and movement distances ranged from about 1,009 to 14,802 kilometers, depending on how long the tags remained attached. The most informative tag was the Gaza-tagged male, which transmitted for 330 days and documented a nearly complete east to west migration cycle. Another important case was tag 112,696, deployed in the Strait of Messina, which lasted 271 days and ended in Lebanon, directly linking central Mediterranean summer habitat with Levantine winter habitat. This figure is based on the Medlem database, with 567 occurrences and provides an important historical context. The pattern is strongly seasonal and geographically structured. In the Levantine Sea, records are dominated by winter observations, while in the northwestern and central Mediterranean, records peak in summer. This independently supports the telemetry finding that the species shifts its distribution across the basin through the year rather than remaining resident in one subregion. Here we see the process tracks of all 9 tagged rays. The strongest demonstration of basin-wide connectivity comes from the Gaza-tagged male, which moved west along the North African coast, reached Spanish waters, then returned east toward the Levant about a year later. Rays tagged in the Strait of Messina generally moved southeast into the Ionian and Adriatic regions, and the longer-lasting Messina tag eventually reached the eastern Levantine Sea. Together, these tracks show that Mediterranean spintail devil rays can connect distant subbasins over seasonal time scales. This slide presents the modeled probability of occurrence by season. The winter prediction highlights high probability in the Levantine and Aegean Seas. In spring, the predicted distribution shifts westward and becomes more widespread, with emphasis on the Adriatic and Central Basin. In summer and autumn, suitable habitat is concentrated more in the central and western Mediterranean, while the Levantine signal becomes weak. This seasonal east-west shift is one of the clearest outcomes of the study. To conclude, this study shows that protecting Mabula in the Mediterranean requires seasonal, basin-wide conservation planning because the same population connects distant regions across the year. Thank you very much for your attention, and I wish you a very successful and fruitful conference.
Steffi's going to be talking to us about her work on the fisheries, trade, and the conservation of manta and devil rays in Peru.
Okay, good afternoon, everyone. Thanks to be here. Today I will present research conducted in Peru. Peru is located in the west coast of South America, facing the Pacific Ocean. If we zoom out what is happening in northern Peru, we can see here that 2 major currents are integrating, the Humboldt current and the El Niño current. So this interaction is creating a highly productive area in northern Peru where the most of the Mobulid fisheries are happening. So around 15 years ago, targeted fisheries for giant manta rays were happening in Peru. Fishers used to say that when mantas rays came close to the coast, they used to catch at least 2 or 3 mantas every single day. Important measurements has been introduced the last decade. In December 2015, national protection was established for Mobula by rostris, and in September 2024, this protection was expanding to include all devil rays. However, the situation is more complex on the ground, And these landings are still, Mobulid fisheries are still happening in northern Peru. So if we see the historical data provided by the government, a total of more than 7,000 tons of Mobulid rays were landing over the 24 years. That means that around 11,000 individuals per year being caught here. The majority of this catch is being by gill nets, followed by pure seine. And if you can see in the second graphic, you can see that the Mobulid fishery is happening all year round, so with some peak in the warm season. We had the opportunity to take measurements of 231 individuals, and the majority of these catches were mobular. And something interesting here is the majority of the individuals were juvenile, so this is something interesting to report. Also, we found gravid mobulids. So we have here to the left Mobula munkiana fetus and a Mobula birostrid fetus. So what is happening in Peru? In Peru, Mobula trade is being being traded locally, and when all the pectoral fins are landing to the fisher terminals, that means that all the head with the gill plates and viscera are discarded at the sea, and all these landings are bringing from the northern to a region called Lambayeque, where all the process is happening. So this meat is being dried here. This is the whole process of the drying. And something important to highlight here is that 60% of the total weight is lost during this process. And you can find the meat in the local markets. You can find specific pectoral fins dried or the meat already shredded. So for Peru, the consumption is really, Mobulid meat are really important both domestically and domestically and culturally important, because we use this meat to prepare a traditional dish called tortilla de raya. And also this species has been represented in pre-Inca cultures. That means that a lot of generations have been consumed these dishes. However, as we have seen, these fisheries operates at a large scale, involve a high proportion of juveniles, and might be impacting important reproductive area. So this highlight the necessity to balance conservation strategy. And just to conclude, I would like to say thank you to all the organizers that is working this work. And if you want to read more about this research, you can use this QR to have the full publication. So yeah, thank you. I don't know.
Yeah, no time for questions. Thank you, Steffi. All right. Last talker before the lunch break, we have Mayuri Chopra. She's going to be talking about her work on Mobulid rays in India, so fewer devil rays in the sea, evidence for declining Mobulid populations off India's coast.
Okay. Hi everyone, I'm going to talk a little bit about, so very briefly, but I'm going to talk about how we generated evidence to say that the Mobulids off India's coast have declined, but also how we identified conservation policies for their population recovery. Okay, so as we know that Mobulids have sadly been declining, and some of the species face declines of up to 99%, and the main cause for that is meat and the gill plate trade. And to address this issue, I'm working in India because it's one of the highly prioritized nations because of the very high bycatch and trade that is sourced from the country.
long story short, so in the last decade, so between 2013 to 2023, I found that there was a significant significant reduction in the mean disc width, which is like a measure of body size, at the population level for Mobula mobular, which is our most dominant species. We have 6 on the southeast coast of India, and this population level decline was, it showed us that there was there was strong evidence of age class truncation in the population, and what that means is that the older and the larger individuals in the populations are declining, or they are being pressured by like fishing pressures, and that's problematic because that is what gives resilience to the population in terms of like when there's anthropogenic stressors. Okay, we also saw very large landings per active vessel declines, which is a proxy of catch per unit effort. So, okay, so we know that the populations are declining, so what do we do about it? So, of course, like the ecologist in me, I made a model. And if anyone wants to talk more about this, come chat to me. And using the population model, I projected the populations forward in different scenarios to see how the populations would respond if we, for example, continued the fishing pressures or if we applied different conservation measures. So this is a scenario where business as usual fisheries pressure would continue for the next 10 years. And interestingly, regardless of the stock size that I started with, because of the population structure that we had, it's very highly likely that the populations are bound to crash if it continues as it is. So like a big takeaway from this is like we definitely need urgent conservation measures. Like even though we don't have the stock assessment, so we don't know like what the population size is, but what we know about the structure is already like giving us this. Okay, so this is sort of what I tried with different policy scenarios. And so, for example, like scenario A and B are size-dependent conservation measures. And each of these scenarios reflect what the conservation measure would sort of bring about, like demographically in the population. So as we see, like all of the scenarios are sort of still not enough for like or as a viable population recovery pathway. So it's only scenario F, which is if we apply maximum bycatch mitigation at the national and the international level, as well as if we have nursery area protection, which gives the space to the species to increase its fecundity. That's literally the only viable conservation management scenario that I found for this population. So yeah, the key takeaways are that, well, the populations are declining quite rapidly annually. And because of these declines, over the past 10 years, we have seen that the population structure has drastically changed, and that no single policy lever is likely going to be enough, so we need integrated management measures which include maximum bycatch protection, but also which include nursery area protection. And of course, none of this would be possible with the wonderful team behind all the works, so thank you so much.
Thank you, Mayuri. That's yeah, scary stuff for the future of that population. So with that, we're already over our timeline, so we're going to break now for lunch. Okay, everyone, welcome to the lunch side event. I'm going to welcome to the stage my colleagues, Nuno and Bex. They're going to be talking today about the new field guide that we've created and a little bit about what it contains, identification, etc. So I'll hand over to them now to lead us from here.
Good afternoon, everybody. Thank you so much for coming to join us on your precious lunch break. It's a real pleasure to be here today representing the Manta Trust. My name is Bex. For those of you that don't know me, I'm the Director of Conservation Programs, and it is a pleasure to be here today along with my colleague Nuno to talk to you a little bit more about Mobulid identification as a tool for conservation management. So before we get stuck into the details of Mobulid identification, I just wanted to give you guys a little bit of an introduction to these incredible species and to give you some context for why Mobulid identification is so important for conservation management. So manta and devil rays are known collectively as mobulids, and we may be a little biased on the Manta Trust team, but we think that these animals are some of the most beautiful, charismatic, and enigmatic creatures in our oceans. So these filter feeders are found throughout the tropical and subtropical oceans of the world. They first appeared in our fossil records around 28 million years ago, and the most famous and iconic of these species, the manta rays, these guys are kind of like the new kids on the block because they evolved only around 5 million years ago. Today, there are 10 recognized species of manta and devil ray, as beautifully illustrated here by Mark Dando. We group these into 3 species of manta, 3 species of oceanic devil ray, and 4 species of pygmy devil ray. And these animals aren't just beautiful and fascinating, they also got the goods up here as well. These animals are highly intelligent. They have the largest brain to body ratio of any species of fish, and the bit of their brain that is associated with the more complex social interactions is highly developed. And some more interesting facts about these creatures. They're incredibly long-lived. So taking mantas, for example, we believe they live to at least 50 years of age. And they also are late to reach sexual maturity. So male mantas reach sexual maturity at around the age of 9, and for females it's even later at around the age of 15 years of age. They also have low fecundity. So they have a very long gestation period of over a year and will give birth to only 1 pup at a time. And this pup will be very large in size at the time that it's born, at over 1 meter in width. And so the females invest a huge amount of energy into growing these enormous pups. It's very taxing on them, and so they can only give birth every 4 to 5 years on average, and in some populations as little as every 7 years. And they also exhibit schooling behavior. Depending on the species, they can be found schooling in dozens, in hundreds, or in the case of the monk's pygmy devil ray, even in their thousands. And unfortunately, this behavior does leave them particularly vulnerable to fisheries. In fact, if we take a step back and look at all of these traits make these animals extremely vulnerable to overexploitation. They simply can't reproduce their numbers quickly enough to deal with even moderate levels of fishing. There are many threats to manta and devil rays, including irresponsible tourism, unsustainable development, and the impacts of the climate crisis. But by far the largest and most acute threat to these animals is that of overfishing. Manta and devil rays are targeted by fisheries. They're also caught as bycatch, and the largest threat to them is actually retained bycatch, which is driven by demand for their gill plates, which are dried and used as an Asian health tonic, but also for their meat. And it was only relatively recently that we started to realize the scale of the mobulid meat trade. Mobulid meat in most places is not considered to be high quality or good to eat, but recent studies have revealed that mobulid meat is being eaten in at least 35 countries around the world. And trade in mobulid meat is primarily domestic, but it is also internationally exported. There are at least 14 countries exporting mobulid gill plates, and these are going to 5 major import hubs, to China, to Hong Kong, to Singapore, South Korea, and to Thailand. And this is where the Manta Trust comes in. The Manta Trust was formed in 2011, to unify and strengthen global research and conservation efforts for these threatened species. We work as what we call an umbrella organization. So at the heart of the Manta Trust is a core operational team that offers support to a global network of affiliated projects. And today we have over 30 affiliate projects that bring together a huge amount of expertise in science, education, media, and policy. And it's really wonderful to see so many members of our affiliate network here today. And we're all working together towards the same shared vision, which is that of a sustainable future for manta and devil rays in healthy, diverse marine ecosystems. Over the last sort of 15 years or so, we have had some important wins in terms of policy and protective legislation for these animals, which you can see listed across the top of the screen. But over the same period of time, we have also continued to see population declines. So you can see here back in 2014, 2 of the Mobulid species were considered to be threatened within the IUCN Red List of threatened species, although several species were still data deficient or not evaluated. But if we jump forward 12 years to 2026, you can see that all 10 species of Mobulid are now considered to be either vulnerable, endangered, or critically endangered. And we are concerned that over the next few years, we're going to see more of these species uplisted as we continue to do further reassessments. And so this is the root of the problem: fisheries. In 2025, a publication on global Mobulid fisheries collated data from the Manta Trust Global Network, and it shows exactly why we're seeing these declines. It highlighted Mobulid fishing to be more widespread and intense than previously thought, with at least 92 countries catching these animals. And it's estimated that over 250,000 individual manta and devil rays are being captured by fisheries each year. Around 85% of this catch is from small vessel fisheries, so we're talking about boats of 15 meters in length or less, and these also account for around 87% of global Mobulid mortality. However, catch from small vessel fisheries is not evenly distributed. The top 5 countries are responsible for around 90% of catches of manta and devil rays in coastal waters, and those countries are Indonesia, India, Myanmar, Peru, and Sri Lanka. And so this is the result of this intense, widespread fishing. We're now seeing declines in these across all species of manta and devil ray in all ocean basins that are steep and continuing to decline. In some cases, we've lost 99% of populations in some locations. And this has all happened within 1 to 2 generations, which is all to say that time is running out. We need to act fast if we're going to save these species from extinction. But there is a glimmer of hope. At the end of 2025, thanks to over 20 years of intense research and hard work by a huge network of scientists and policymakers, we were able to get all Mobulid species uplisted to CITES Appendix 1, giving them the highest level of international protection in trade. So this now gives us an amazing opportunity to finally get protections working for Mobulids around the world and to start to tackle the trade in their gill plates and in their meat. So the Manta Trust's priorities following this exciting victory are to prioritize the following. We want to see greater national protections for these species in priority countries. We're working to improve enforcement capacity to actually uphold CITES legislation and national protections. We are gaining a greater knowledge of the trade and trying to make it traceable. And we're also working hard to reduce bycatch mortality. So if anybody in the room is working on any of these things, we'd love to speak to you. Please come and find someone from the Manta Trust team this week so we can chat to you more about it. To help us fill critical knowledge gaps to support this work, we really need to improve both the quantity and the quality of data that we're receiving on these animals. One of the problems we have currently is much of the catch data we receive is labeled simply as Mobula, which just doesn't give us the information that we need to fully understand the impact and to work on tailored conservation strategies across these different species. Now, the problem is identifying mobulids can be really tricky for lots of reasons. There's a lot of visual similarities across the different species, making them hard for non-experts to understand what they're looking at. Even within a single species, there's highly variable patterning and coloring on these animals. They also make life extra difficult by changing color and losing some in their patterning after death. And also really important to bear in mind that we're often not working with whole specimens, rather we're just working with body parts. So these are our data collection challenges, and this is why mobulid identification is so important for conservation. And this is where the Manta Trust new field guide to manta and devil rays of the world comes in. And it gives me pleasure to invite my colleague and one of the co-authors of this field guide to the stage. Nuno's now going to tell us a little bit more about the guide, and he's going to show us how to identify a mobulid.
Thank you, Bex. It is now my job, as Bex said, to take you on the ride to the inside of this guide. And starting with who made it. So the book was put together by this selection of mobulid-loving humans that you have over there. Guy Stevens, myself, Betty Lagelbauer as the Manta Trust's contingent of the authors. The amazing Mark Dando, Shark and Ray illustrator, who may or may be not hiding in the back somewhere, hoping I didn't say his name out loud. But he's the reason why the illustrations featured in this book are both visually stunning and scientifically accurate. And also two big names in mobulid conservation, our hosts, Daniel Fernando and Giuseppe Nortarbartolo di Schiara. Shocking, I believe I got Giuseppe's name right. Because we had to tackle all the ID challenges that Bex just mentioned, we populated this book with as much pictures, illustrations, maps, and plates as we can. This is a book that is not meant to be collecting dust somewhere in a shelf. So it's a book made to be in the field, fish markets, customs offices, landing sites, and even other types of challenging environments, such as the desks of policymakers. I had to say that one. This is why it's made with durable paper, and it's ring-bound so you can easily flick through it, and it's available in 4 languages, for now, hopefully. Portuguese, Spanish, French, and English. Now, I won't have time to actually give you a proper tour of the book. Let's just say that there is new things in it and extensively revised things in it. So, in the new things, there are new illustrations, plates, post-mortem color changes. This is very important because all the illustrations that were featured, all the mobulid illustrations featured in guidebooks so far were based on live animals, and this wasn't necessarily very helpful in identifying dead specimens in fish markets and landing sites, because what happens is when a mobulid dies, the pigmentation contained in the mucus coating is removed, gets rubbed off, and what happens is they change their colors and their patterns. They're usually just darker, and the patterns look fainter. And for this reason, we collected the most number of pictures that we had from all around the world to cover regional variations, to then hand over to Mark for him to make single species illustrations that would more or less be standardized per species. Another new feature is the safe handling and release guidelines. So this didn't come off of the top of our heads. We put together a wide group of experts per gear. Many of them are here. And to try to reach simple, clear instructions and recommendations per gear type, so per seine, long line, and gill nets, and also what not to do's that would cover all these gear, and this is featured in the guide too. And it's already making its way into international policy, thankfully. Also, other sections were extensively updated and revised, such as taxonomy and species accounts, ecology, distributions, and threats, because there's a lot of new science out there. Because this is an ID guide, the main focus is in knowing which species we're talking about. This is why we put a lot of effort into key diagnostic characteristics, ventrally, dorsally, laterally, and also we revised the ID keys for both species and gill plates. But we don't just need to correctly pin down our species, we need to learn how to start narrowing down our search, and this is why we also put some effort into secondary ID characteristics, such as behavior, feeding strategies, breaching behavior, aerial ID, because there's a lot of people with drones out there, and also associated species of fish. And there is also in the book a proposal for standardized data collection protocols, because it's very important that we're all collecting the same data in the same way. Now, enough about the book, and let's get into the fun stuff. The book is meant to ID mobulids, so that's what we're going to do right now. So let's start with a practical exercise. We are out in the ocean, and we see a ray. We think it might be a mobulid, but we can't know for sure. So what we need to do is we need to start asking ourselves questions. But it can't just be any questions. It has to be the right questions in the right order. So the first question is, obviously, is this a mobulid? And to answer this, you need to actually look at the shape of their heads. So other rays that can be confused with mobulids have either a round or pointed snout, or a broad, flattened snout that extends beyond the head. But only mobulids, manta and devil rays, actually have cephalic fins. Now these structures, when furled up, resemble little horns, and hence the term devil rays. When they are unfurled, they act as pretty much paddles that funnel the plankton-rich water inside their mouths for feeding. So, question number one, is this a mobulid? Yes, it is, because it has cephalic fins. First question out of the way, we move to question number two. Is this a manta or a devil ray? And for this, we need to start by looking at their mouths. Now, this is quite easy. Manta rays have a terminal mouth, which means that jaws are aligned, as devil rays have what we call a subterminal mouth, meaning that the bottom jaw is undercut. So it's actually facing in the ventral side of the animal. Question number two, is this a manta or a devil ray? And the answer is, it is a manta ray, because the jaws are aligned. Now, before we get into all the markings and all that stuff, we need to narrow things down. And because these things can't be easy, there's 3 species of manta rays. So one of the things that we can start asking ourselves, and that even regardless of the quality of the observation, one thing that we may know for sure is where we are. Because not all these species swim in the same waters, and we know for sure that that ray that we saw when we were diving, for example, in the Indian Ocean. This already helps us, because this rules down Atlantic manta, which is bound to the Atlantic Ocean. So now we are in a situation that is more comfortable for us, which is what we want to be, which is on a one-to-one face-off between 2 species that we will analyze. So, question number 4. Let's dive in to see what markings these animals have. So, on oceanic mantas, dorsally, there's white triangular markings that create this kind of T-shape, this black T-shape in the middle. In reef mantas, that's not the case. So these white markings are not so sharp, they're more faded, and the T is not a T, it's more like a V. When we analyze it ventrally, you see that there's even more differences. For example, the oceanic mantas are usually shaded black in the inside of their mouths and cephalic fins and trailing edge of the pectoral fins, which is not the case in reef mantas. There's also black shading in the gill covers, especially the 5th gill, and there's not so much in the reef manta. And the spots, obviously the spots. In the oceanic mantas, the spots are usually clustered in the abdominal region, and in the reefs, they're usually between gill slits and spread across the trailing edge of the pectoral fins and abdominal region. So, the question is, which species is it? What shall it be? It has white cephalic fins on the inside, a V-shaped black pattern in the back, which means we are in the presence of a lovely reef manta. Now, what we've just been through here, starting from the very beginning and knowing that we only started with array, is basically what the guide makes you do. So this little comparison that we just did is essentially going through the species
Whoa. Sorry, Nuno, I just need to interrupt.
Please do.
I thought we were trying to impress these guys.
Well, I was.
Okay, that was way too easy. We did a much harder example to show you guys how good this process is. We talked earlier about there being some challenges with identifying mobulids. So things like, we might not know where the picture was taken. We also might not have a great viewpoint and be able to see all of the animal, to look at their markings or some of these things that Nuno's already mentioned. We might be working with a dead specimen, and we might just be working with a body part. So I'm going to do something way more impressive than what Nuno just did. I'm going to identify this dead mobulid. Here we go. So, question number 1, is it a manta or a devil ray? If we take a close-up look at its head, we can see that the top jaw is overbiting, so we've got ourselves a devil ray of some kind. Moving on quickly. There are 7 species of devil ray. So one of the things we can look at is the size of the animal to give us a clue. All of the oceanic devil rays grow to over 140 centimeters in width. So if we have one over that size, we have an oceanic. If it's smaller, then it might be a pygmy devil ray, or it could be a juvenile oceanic devil ray. Now, luckily, in this picture, we have somebody in there for scale, and we can see that this animal is probably over 140 centimeters. So that helps us to narrow it down to one of these 3 species. So the next thing we want to look at is their color and markings. But as Nuno's already mentioned, this is a dead animal. So we need to switch up looking at these images of the live animals and switch to looking at images of the dead animals, which are all contained within the field guide. And if we take a look at these 3 and look at the image of our dead mobulid, we can quite quickly rule out the sickle fin devil ray, which is quite brown in color, whereas the image we're looking at looks very dark and black. So I think it's probably a spine tail or a bent fin devil ray. But to guess which one it is, because those markings on the animal seem to be missing, we're going to have to take a closer look and take a look at its head. Now, if you look at these images of a spine-tailed devil ray's head and a bent-fin devil ray's head, you can instantly see some quite big differences in their markings. So spine-tailed devil rays have this white bit that wraps up behind their eyes, whereas bent-fin devil rays, that white bit doesn't go up any higher above the eye line. Spine-tailed devil rays also have a much darker eyeball than a bent-fin devil ray. And another clue you can look for is where the spiracle sits. So on spine-tailed devil rays, the spiracle will sit above the margin where the pectoral fin meets the body, whereas on a bent-fin devil ray, the spiracle sits beneath this line. So if we go back and take a close-up look at our dead mobulid, it's not a great shot, but we can just see enough to see those white markings coming up and above the eye line and wrapping up behind the back of the eye. And we can also see that the spiracle sits above the margin where the pectoral fin meets the body. And so all of that means we are looking at a spine-tailed devil ray. I would say mic drop, but I can't drop these because they're attached to the table.
All right, smarty pants. I admit, that was, kind of neat. But, anyone can identify a whole dead mobulid. You want to know how it's done? Yeah. All right, let's move on. Mobulids are not always a whole. What if you just have a dried gill plate? That is even much harder because it poses additional challenges. It can't even be classified at the species level many times because, as you can see, they're all different, but you can only group them into 4 categories. So 3 indistinct species of manta ray, Sickle-fin devil ray, spine-tailed devil ray, and all other mobulids, which means all the pygmy devil rays plus the bent-fin devil ray. So for this, we created the gill plate ID key, and this is based on 3 basic characteristics that every gill plate has, which is size, color, and lobe edging. So size can be either small, medium, or large, color, uniform or bicolored, lobe edging, smooth versus jagged. So you're strolling down a fish market. You come across this dried gill plate. You think it's mobulid, but you can't know for sure which species it is. So you start going down the gill plate ID key. And the first question is this gill plate longer than 25 centimeters and without a distinct bicolored pattern? Well, the size doesn't look like it's longer than 25 centimeters, but let's look at the other characteristic. It's definitely not all black, as it states there, because if it would be a Manta gill plate, one of the 3 species. So we move on to question 2. Does it have a thick white center and smooth lobe edging? Well, I guess it does. It does have a thick white center. Those lobes kind of seem smooth. We have a winner. It's a sickle-fin devil ray gill plate. If we wouldn't have a winner, what we would have is question number 3. Does it have jagged and white lobe edging only? If it's a yes, then it will be a spine-tailed devil ray, mobular gill plate. If not, then we are down to the other half of the species of mountain devil rays, which are the pygmies or mobular thurstoni. What do you think? We give this a lot a go?
Yeah, they look really smug. I've seen a lot of people laughing at us, and I think we'd like to see how good you guys are at mobulid identification now.
All right, let's give it a shot then. Because we won't have the time or space to do really fancy stuff like brainstorming sessions and grouping you into little pairs and things like that. The way it's going to work is we're going to put a picture out there with 2 options. And because there's not so many of us, how it's going to work is if you believe it's option 1, we'll call you to cheer and make yourselves heard, stand up, dance, sing, whatever. Just make yourselves heard. If you think it's option 2, do the same thing. Now let's go for the first round. Is this a manta ray or a devil ray? If you think this is a manta ray, make yourselves heard. Cheer now. Come on, guys. I know you didn't have lunch, but come on, give us something. If you think it's a manta ray. Obviously, if you don't. Let's hear it for Team Devil Ray. Guess what? You were right. And you know why? Bottom jaw undercut. Section 2. Is this an oceanic or a reef manta? If you think it's an oceanic, make yourselves heard now. Don't think we can fool these guys.
Yeah, we're going to have to make this harder.
Is it a reef manta? Team reef manta? Silence. We tried to put the reef picture there to trick you, but it's still an oceanic devil ray. Black shading inside of mouth, 5th gill cover, and clustered spots in the abdominal region.
Right, let's try and make this a little more challenging. Here's a dead one. Okay. Let's hear it for spine-tailed devil ray. And Benfin? Oh, you guys are too good. Yeah, we got the white markings wrapping up behind the eye, the spiracle above the pectoral fin. All right, let's try and amp things up a little further. We want you to identify this gill plate, the one in the middle with the big orange arrow pointing at it. So, after 3, if you think it's a spine-tailed devil ray, cheer. 1, 2, 3. And if it's a manta ray? Yeah, well done, guys.
All right, I think these guys are experts by now.
Yeah?
Yeah. All right. So let's go a little bit off script. Let's talk about something that we actually haven't talked about in this presentation. I mean, slightly, because we have said that location sometimes narrows things down. So imagine this. you can not have a good view of the manta or the devil ray. You can just have just a glimpse of a shadow, but you still think it's a mobulid. You're on vacations, let's say in Greece. Here it is, beautiful Mediterranean landscape. You see a mobulid, or at least something that looks like a mobulid. What is it? If you think it's a spine-tailed devil ray, cheer now. If you think it's a reef manta, cheer now. Yay, we nailed them. It's a spine-tailed devil ray. And you know why? Because this is the only species present in the Mediterranean Ocean. I know we haven't talked, wait, I know what you're going to say. Up until last month, because the first record of oceanic manta was published for the southern coast of Spain. But that doesn't matter because oceanics is not an option here, okay? You think we can give them one more?
Yeah, all right. We're going to give you one with no clues, just to be really cruel. You're going to have to shout really loud. Does anybody here think they might know what species this is? On a count of 3. 1, 2, 3. I heard pygmy. I need the species. It's a monk's pygmy devil ray. These are the gorgeous little guys that we heard Marta talking about this morning, and they can be easily recognized when you get them in these enormous aggregations, which can be in their thousands. So even from an aerial shot like this, where you can't see any of those features, you might still be able to identify them purely through their behavior.
All right, that was fun. But we think there's still one question, although we know all this. We're all mobulid experts by now, gill plates, body parts, whole animals, live, dead. But there's still one thing that we need to address. Why? Why do we want to become mobulid experts? What does it mean? I mean, what is it for? And this is where conservation comes into play, and where mobulid ID acts as a tool for enforcement of current protections. Now, when we were putting together all that data that Bex was talking about in the beginning, all that species-specific trends, one of the main constraints that we found was actually in the data itself. First of all, because only a fraction of the data of the global catch, the real one, actually gets reported to the official databases, and that's how much of a fraction it is right there, just a glimpse of it. And when it does get reported to databases such as the FAO or to NRFMOs, again, like Beck said, it gets aggregated into mobulids or just manta or just random names like that, but almost never at species-specific level. And also, it gets reported in weight units, such as tons. Now, if you know that somewhere someone caught 10 tons of mobulids, that's not going to give you much data for science and for species-specific population structure. Along the data, we found also many inconsistencies and taxonomical misidentifications and taxonomical inconsistencies that continue to perpetuate in the data. So we need to improve species-specific data collection. I don't know if we mentioned this, but we made a book which can help in this. One of the other things that can help is the standardized data collection protocols in the book. Because again, we all need to be collecting the same data in the same way. Also advocating wherever and whenever we can for species-specific data collection in institutions such as NRFMOs. And we need to convert these weight into individual mobulids. And for that, we are working on 2 key publications. One is the size-weight ratios paper and the body weight measurements, body part weight measurements paper to try to convert these units, these weight units into individual mobulids per species. Now, when you look at the trade data, the same patterns emerge that when we were looking at the fisheries data. Only a fraction of the actual trade actually gets reported, as the recent reassessment of mobulid meat and gill plate consumption and trade, led by Marta, also indicated. Now, we know that there's more than 25 countries involved in international trade in mobulid products. Of these, only 7 are featured in the Society's Trade database for the whole period where these animals were Appendix 2. And of the volume of trade reported for all these years, 75% of it is just between two countries, Sri Lanka and Hong Kong. So most of it is just unreported and therefore possibly, most likely, illegal. No mention of meat trade, because this is even harder to track, and we know that it gets exported from at least 10 countries into another 5. Coding errors are highly suspected when you analyze all this data, and again, it's classified and listed as mobulids and not species-specific. This is just to show that along the whole fisheries and trade chains, there are very poor identification skills. And this needs to change if we want to have enforcement of the existing current protections and if we want to have tackling of illegal trade. We need everyone in customs offices, fish markets, landing sites, to very accurately and swiftly know the answer to 2 very simple but very important questions. What is this? Is this a mobulid, what I have in front of me? Is it a whole, even if it's a whole mobulid, piece of meat, gill plate, and which species population does it belong to? For this, we need tools. We have a guide, but we need to go further, because not everyone has the guide. The guide can't be everywhere, but everybody has a phone. So what we are aiming to do, and the 2 tools that the Manta Trust is starting to develop right now, the first one is the Mobulid ID app that will have an integrated gill plate photo ID software. This is an interactive tool based on the guide. It will also be a citizen science platform where everyone can submit pictures. And again, the gill plate ID software is being developed and pictures are being collected to feed this technology. This will be step 1. Step 2 is the Rapid Genetic Toolkit. So this tool is to quickly ID mobulids at fish markets, customs offices, etc. Our genetics team, which is also here, has successfully created genetic primers to amplify the diagnostic genome of mobulids, now in laboratory conditions, but hopefully being tested in the field quite soon. This will provide info on species and population locations for species where this is feasible and that are present in more than one ocean basin. And people need to know how to use these tools. So we will also have to produce enforcement resources such as training videos, etc., to make sure that these tools get applied by the right people in the right way. All of this, when in place, will then allow for the existence of a protocol when something looks fishy. The protocol will be something like this. A shipping or landing looks suspicious. Using the app or the guide, officials will be able to confirm presence of mobulids using the app or presence of mobulid gill plates using the photo ID software within the app. This will then trigger that the shipping or landing will be held for investigation. And because the genetics work needs to be done locally in partnership with local labs, then samples need to be taken to local labs to be analyzed using the Rapid Genetic Toolkit. And hopefully within 1 or 2 days, confirm the species of mobulid, and when feasible, confirm the geographical origin. This will then constitute proof. Proof that can lead to seizures, fines, prosecution, tackling of the illegal trade, because CITES itself is not a miracle. It needs to be enforced, and there needs to be tools for that. Now, I don't know if I mentioned this before, but we made a book. No, I'm just kidding. The book is available at nhbs.com, the distributors of the book, in each of the 4 languages. It will be an interactive ePDF soon, and if you work in the field with mobulids, if you think the book can make a difference for conservation through your work, we still have very few books here left. So if you think that you are entitled to one, please come and find us, and hopefully we can have one to spare. Any questions can be addressed to either myself, Bex, or Guy Stevens, the main author of the book, CEO of the Manta Trust and moderator of this session.
I have a question, Nuno. Actually, I'm going to ask Bex. What species is that, Bex?
These are, it's monks. Monks
Yes.
could be devil ray.
I'll get you out.
That was close.
All right, thank you very much, you two. Does anyone have any questions? Anything you would like to ask the team, or come down and present your case for why Nuno should hand out one of the guidebooks to you guys? Yes, and we're also around here for probably the next 20 minutes until the session starts. So yeah, please come and speak to us. Otherwise, thank you very much for coming. We appreciate it very much. Thank you. All right, so first up to the stage, we have Alejandra Arana. She's going to be talking about genetic tools for tracing the global gillplate trade in critically endangered devil rays.
Hello, everyone. Thank you for coming. I hope you are not asleep after a very good lunch. Well, the international gillplate trade remains one of the most significant threats to mobulids. The recent uplisting to CITES Appendix 1 is a historic victory for conservation. Reproduction in paper isn't enough. Now more than ever, what we need are powerful, practical tools that can support trade monitoring and enforcement. Even with all mobulids now on CITES, enforcement often happens at a species level. So, species identification is still important. This can be a challenge, even with molecular tools, because the gillplate products, the way they are dried, the way they are processed, damages DNA. To address this, we have designed a mini DNA barcode that is able to distinguish between the main traded species using a very short fragment of DNA, so short that it can be used in the most damage of samples. But species identification alone isn't enough. We know that the gillplate trade is rapidly evolving. It's going online and moving faster than ever. We need intelligence gathering tools that can help us understand how this trade is expanding. Which brought us to our next big question. Can we use genetic information to identify where these gillplate products are coming from? In my PhD, I focus on two critically endangered species, the spine-tailed devil ray and the bent-field devil ray. These are highly traded species and have a circumglobal distribution. I use genome-wide data to assess the connectivity between populations across their global range. And what we found is exciting. We found clear genetic differentiation between populations in the Indo-Pacific region and the Atlantic region for both the species. This is exciting because it's the first time that we have this kind of analysis at a global scale, but what is more exciting is that this means that genetic geographic tracing is possible. So building on these results and previous genetic work on other mobulids, we are doing this. We are developing a set of genetic markers for geographic tracing of mobulids with geographic signal. Our workflow basically has three main stages: computational design, lab validation with muscle tissues, and field testing in major trade hubs using gillplates. As we move into the first stage, the design, we keep asking ourselves this very simple but key question: how can we make these tools actually useful for people in the front lines? So we're assessing a wide variety of methods, from rapid tests, like the COVID ones, to more complex sequencing, taking into account the genetic resolution, the cost, the speed, but also being very aware of the practicalities of costumes and the different contexts in different countries in terms of their forensic labs. Because ultimately, what we want to do is to design a reliable species ID and geographic tracing tool that can empower local stakeholders to turn CITES protection into real-world enforcement. You can help us achieve this. We need more muscle tissue samples from the Atlantic, and we would love to have more collaborators to coordinate in-country pilots in key trade hubs. I hope you approach and we connect, because saving these Manta and devil rays is not just about making science that sounds exciting. It's about making science useful across borders. Thank you.
Thank you, Ali. That was great. We have time for a question or two, if anyone would like to ask Ali something. We have one. Jessica, you're up again.
Well, I was just curious about why you think the Atlantic and the Indo-Pacific are different populations. And I'm just thinking of it from the manta perspective as like the reef mantas are Indo-Pacific and third species is Atlantic. Is there something related there?
I think it's just in terms of how the geographic ranges overlap. They need temperate to warm waters. And so, yes, it's just the way like the geographic ranges work. I don't think there's any other reason. Yeah.
Thank you, Ali.
Thank you.
Next up, we have Christina Ekanayaka. Sorry, I didn't get that right. Evaluating the robustness of length-based Bayesian biomass model to determine the stock status of Mobula mobula in Sri Lanka.
All right. Hi, everyone. My name is Christina, and I will be sharing with you just a snippet of my undergraduate research that I did in collaboration with Blue Resources Trust. So my project aimed to assess the stock population of the spine-tailed devil ray, or Mobula mobula, here in Sri Lanka. But first, a little bit about the species. As we know, they are critically endangered and highly exploited, especially for their gill plates. A not so fun fact, Sri Lanka's artisanal fisheries actually have the highest Mobulid captures compared to all of the globe's purse seine fisheries combined. Now that's a lot for a small island. In Sri Lanka, there isn't a lot of data available for stock assessments. So I used a data-limited approach that utilizes length measurements. The data that I used in the study was collected by Blue Resources Trust, where 77 landing sites were visited, and the disk width of 919 landed Mobula mobula individuals was measured. All of this data was then fed into a length-based Bayesian biomass model that I modified to fit the data that I had. Now, this model uses the species' biological information called priors to infer if the population is overfished. But what I found was, as I changed the values of these priors, my results changed with it, quite drastically, which is a little unusual. So I conducted a sensitivity analysis to figure out just how much these results were influenced by the priors. One of the priors that I looked at was called asymptotic length, or L infinity, which refers to the maximum length a species can reach. I tested a range of L infinities from 250 to 350 centimeters at 10 centimeter intervals. This range was chosen because 250 centimeters was the largest measurement I had in my data set, and 350 is actually the published L infinity for the species. What I found, though, was that the model actually did not converge with the higher values of this range. And this is likely due to the fact that there was a lack of larger individuals in my data set. As I mentioned before, the largest individual I had was 250 centimeters wide. So this actually points to the presence of growth overfishing. For all the values that did work, however, the results showed overfishing. So what does all this mean? It's the big question. So based on my results, we can say that the current L infinity for the Mobula mobula population here in Sri Lanka is much lower than the published value. This essentially means that this population is so overfished that they can't even reach their maximum length. So in conclusion, we can say that Mobula mobula in Sri Lanka is overfished, and my study also highlights the importance of reliable, up-to-date biological information that can help stock assessments and also help to manage fisheries sustainably. If you're a little curious about the other priors I looked at, please come and find me after, and I would love to share. I just want to say thank you to the BRT team, both past and present, Deakin University, my supervisors, and OCS for sponsoring my travel, and thank you for listening.
Thank you, Christina. We have time for a question. I have one. Well, it's more of an observation, really. Is I had similar discussions with my colleague Nidhi recently about this considerable variation in the catch max landings for this species in the Indo-Pacific versus the Mediterranean, where you have these much larger reported individuals. She was trying to figure out how to address that in some of the publications that we have at the moment. So I think the two of you should definitely
yes.
talk about that.
Okay.
Love to speak to you about that.
Yeah, definitely. Thank you.
Thank you very much. Okay. Last but not least, we have Gigi Muller. Gigi, welcome to the stage. Gigi's going to be talking about the reproductive ecology of reef manta rays on Australia's east coast.
Thank you for the introduction, Guy. As you already said, my name is Gigi. I studied the reproductive ecology of reef manta rays on Australia's east coast using long-term photo ID data. As most people in this room would know, our understanding of the reproduction of manta rays is still quite limited, with only really a handful of publications about their fecundity from wild animals from a few different regions, all suggesting sort of high variability. So that means that population-specific data are critical, and here I'm presenting a first assessment from Australia. To assess fecundity, we used 15 years of photo ID data, the Project Manta database, and that was nearly 8,000 sightings from over 1,200 individuals. And we identified pregnancies looking at the ventral and dorsal distension that becomes evident in later term pregnancies. We focused on two key aggregation sites, Lady Elliot Island and North Stradbroke Island, which Asia has already introduced in her talk. And we have high, or like relatively reliable re-sightings of individuals at those locations. Thank you. Briefly, for context, while reef manta rays are globally classified as vulnerable, this population appears to be relatively stable, so it's a really great place to study their reproduction. Across our data set, we recorded 249 pregnancies from 169 females, so about a quarter of our females were seen pregnant throughout the study period. Consistent with other studies, we recorded high interspecific variation. 49 of our females were seen with multiple pregnancies, with a maximum of 4 pregnancies across the 15 years. Some were seen pregnant in consecutive years, while others, even with annual sightings, had big gaps in between pregnancies. In addition, our data showed clear seasonal trends at the two aggregation sites. If you look at the circular plots, on the top you see Lady Elliot Island, where we saw heavily pregnant females during Australian winter. And then down south at North Stradbroke Island, we saw heavily pregnant females again, but during Australian summer. Similar trends have been noticed in previous studies from different locations, and that suggests that manta rays may have a partially defined annual reproductive cycle, which essentially means that they may have multiple ovulations throughout the year, so multiple opportunities to reproduce. When we focused on high re-site individuals only, the average reproductive interval was 1 pup every 5.8 to 8.8 years. That's very little. So if we take the sort of estimated lifespan of 50 years and age at sexual maturity for females of 10 to 15 years, that would be 1 pup, that would be 4 to 7 pups throughout the life of a female. And previous demographic models have used much more optimistic reproductive parameters, with like 1 pup every 2 years. We know from the IUCN assessment already that a more conservative estimate is probably better for the species. So when we plotted our reproductive parameters in those demographic models to estimate maximum population growth rate, RMAX, we got similar values to the IUCN assessment, and that would be a 2 to 3% growth per year, and that is under ideal conditions. That's sort of the framework of the model. And bear in mind, this is coming from a population where we don't really have direct threats. They're not targeted for anything. So what's next? This project was part of my master's thesis, and while long-term photo ID data provides really valuable population-level insights, the method has limitations for reproductive studies, as you can imagine. So as part of my PhD, which I'm starting later this year, I'm going to be incorporating ultrasound and hormone analysis to better understand the species reproduction. We've already conducted a pilot field trip to Lady Elliot Island together with our collaborators from Okinawa Shirashima Foundation. You see Kiyomi, Queen of Ultrasound, ultrasounding a female, so I'm really excited to continue this work. Oh no. Thank you, of course, to my supervisors, Asia von Project Manta, Mark Erdman from Rewild, BioPixel Oceans Foundation, thanks Sharks International for having me. If you have questions, please find me. If you have done hormone analysis for manta rays, please also find me. Thank you.
Thank you, Gigi. Yes, I need to connect you with Beth because she's already done it.
Amazing.
So, yeah. And I've got a copy of the book for you, too.
Cool.
All right. So that wraps up the Matters session. Thank you, everyone, for your attention.
Filippo Bocci with tiger sharks.
Afternoon, everyone. My name is Filippo. As already been announced, today I have the pleasure of introducing you to an effort of local NGOs, dive center, and international institution to identify the world's largest aggregation of tiger sharks in Fuvahmulah, Maldives. So some of my colleagues have already been talking about Fuvahmulah, but for those who weren't here, Fuvahmulah is an oceanic atoll located in the southern side of the Maldivian archipelago, and there has been a dedicated effort of photo identification in roughly 5 years to identify the world's largest aggregation. We have been conducting daily diving there, and we've discovered a population of over 300 individuals. Most of them are females, although males do seasonally occur at the dive site as well. The power of the photo identification in this sense is that we were able to track the abdominal distensions of the pregnant or what we supposed to be pregnant females, and also to track the departure times and leaving of and returning of the pregnant females. And we managed to connect that with a potential asynchronous reproduction because peaks in absence were essentially in 2 different periods, from December to January and from July to August. We further investigated into that, looking at what it could be the potential cycle of the reproductive females. So essentially we managed to understand that there were 42 periods of this absence time that roughly correspond to circa 25 months, which is what is closer to a biannual cycle in the area. But to further validate our photo identification data, we decided to look further, even more into this by using an underwater ultrasound, which is a device that we use underwater while we were diving, totally invasive for the female. And we managed to understand that our assumptions were right because the females were there at the same time, but having the pups have 2 different stages of development. And we statistically significant found a difference. We tested and we found 2 different types of early term and mid-term group of females. Now, you might be wondering what our future directions are. So we obviously have a lot of questions. Some of my colleagues in other research areas have tried to answer to some of them. But when it comes to us, one of the main questions is now to understand what could be the important gestation ground of these females. Obviously, we have a pretty good understanding that Fuvahmulah acts as a gestation ground. But once these females leave the water of Fuvahmulah, we don't really have an idea where they currently go. So identify the parturition sites and nursery ground is the next key step. We also want to have an understanding of their movements, implying satellite tags, and probably looking into genetic studies as well as diet and how could they impact the provisioning. Thank you so much, Shark International, for having us here. And I want to particularly thank you, our contributor financially, Ina, when it comes to our NGO and Dive Center, and Tatiana and Ibrahim, which are also sitting there with Fuvahmulah Dive School, for believing us a researcher. And we want to obviously highlight also the huge help of the locals in keeping us safe and diving with us, and most probably in the future they will be involved in the research operation as well. Thank you very much. I will take any questions if I have time. Otherwise, I'm sticking out at the conference, or you can just email me as well.
Any questions?
There's one over there, I think. Nico?
Hi, Filippo, epic talk. My question was, have you guys thought about using birth tags to see, you got these pregnant sharks around, but where do they actually go and give birth? So, the ones that are outside at wildlife computers?
Yeah, sure. It's definitely an ongoing conversation. We have looked into that. Now we are still trying to understand the impact that this type of tags could have, but for sure, it's something that we vouch as an option to look into for having an understanding of the research question that I just display on the side. So for sure, that is on the list.
Thank you, Filippo.
Yeah, thank you so much. All right, thank you.
Okay, thank you very much, Filippo. Next up, we have Maite Erauskin-Extramiana.
Yeah, thank you. Good afternoon, everybody. My name is Maite, and I come from ASTI, and I would like to have a little and fast overview of the project that we are doing there. So basically, each dot is a presence of blue shark. I'm from here, so the question was why they like this area. But surprisingly, there was nobody studying the sharks there. So we proposed a project 4 years ago, and we took a multidisciplinary, interdisciplinary approach. So we started analyzing fisheries data because there was a fleet, long-line fleet targeting blue sharks in the area. We also worked together with captains of those longliners to get their knowledge of the areas, the seasons, the size of the species that we were working with. We started tagging with satellite tagging and acoustic tagging those animals. And last year we got the OTN loan to deploy the first receivers in the Bay of Biscay. You can see the gap the ETN has in the area, so we can start trying to fill this gap in the area. We also collaborate closely with a group that is deploying BRAVs and doing eDNA analysis. And we start doing also landing samplings at port because they land those blue sharks, and to see the sizes, the sexes, and the structure of the population that they were catching. And we include everything in the habitat model with the aim of overlap with different marine activities and identify these hotspots, potential bycatch hotspots. We made a citizen science initiative, and thanks to that, we were able not only to get blue shark reports or catch tag blue shark, but also porbeagles, basking shark, surfing mako, hammerheads, and also some fishermen reported white sharks. So in the left, you can see the tagged animal patterns. We thought at the beginning that they just came through the Bay of Biscay in the way from Ireland to Azores, but then we saw that they spent more time than we thought at the beginning, and we found that we have an interesting pelagic shark community there. We also were able to tag the first mako shark in the Bay of Biscay, and we saw that she spent a lot of time in the Capbreton Canyon, then she went to Portuguese canyons, and then she went straight to the Cabo Verde, and when she started going up again, she was fished, although there is a ban for this species. In our field trips, we were able also to film the first mating attempt between blue sharks, and also for the pelagic extreme race, so all these insights gave us the idea that maybe the Bay of Biscay can be potentially a nursery and reproduction area. So we also have a lot of females with mating scars, very small individuals. This one is not the smallest one, and due to the citizen science, we got pictures for super small porbeagles and surfing mako sharks. So this project is finishing next year, but we would like to move on to try to answer if this area is a nursery area and also a mating or reproduction area, and move on tagging acoustically, start seeing the detections, because we have some sharks detected in Galicia and in France. Move on deploying BRAVs, eDNA, and of course bring all this knowledge that we are creating to the management authorities at ICAT. So thank you very much, and if you have any questions, please reach me out and we can talk further. Thank you.
Got time for one question? No?
Thank you. Yeah.
Thank you, Maite. That was such a great talk. I'm just wondering about the management side of things. Sorry, the echo is like so hard. Yeah, like what are some of the recommendations you might make to ICAT that would reduce pressure? I mean, it was interesting to hear that one got fished. Yeah, any thoughts on that?
So for example, which is very interesting when we work with the long-leg captains is they don't want to fish the small individuals because they need to remove them. For them, it's not interesting at all, and they know the importance of having them to have sharks in the future. So if we are able to manage, especially or seasonally, these areas that they are identifying and we can model that can be of interest, maybe to tell them or maybe to have recommendations of don't fish in this area of this season and yeah, try to avoid it. So the spatio-temporal closures or recommendations or something like that. I think something in this way. Thank you so much.
Okay, next up we have Muhammad Hanif Fadzli on growth and age parameters of bamboo shark.
Okay, hello everyone. My name is Hanif from Malaysia. Today I'm going to share to you about my study on the age and growth parameter of Cirrus putatum from the Malaysia. Cirrus putatum is one of the species that have been highly landed in fish east coast of Peninsular Malaysia. This species was, however, we doesn't know the biological data such as age and growth parameter, and this species was vulnerable towards fish such as fish trap and fish trawl. In past previous study, we know that this species is a slow growth rate and low maturity and low fertility. However, in Malaysia, we doesn't know. So in this study, we would like to focus into two objectives, which is to determine the age structure and estimate the growth parameter. The sample was collected at the east coast of Peninsular Malaysia from Kuala Dungun and Kuantan Fish Landing Port. Then the sample has been taken the measurement, weight and the sex. After that, the vertebrae was extracted. Then we clean it and burn it using the protocol by Fahmi et al. 2031. Then we estimate the age by observing the opaque and translucent zone. For the model, we choose the volumetric growth model to calculate the growth parameter. For the result, we managed to get that the composition of age for Cirrus putatum from the east coast of Peninsular Malaysia were from 3 years old to 10 years old, where the males has higher mean at age 7 and female was age 6. And for the growth parameter, we have observed that the female has large aseptic growth, and however, the growth rate is a little bit slower and contradict towards the female. Female has higher growth rate and low aseptic growth. We assume that this species has sexual dimorphism characteristic towards their growth. If I talk this, it's going to be long, so I'm going to simplify into 3 main points, which I would like to suggest that enforcing the minimum size limit towards this species and protecting the immature individual by releasing it. Also, I would like to recommend for implementing area of closure, because during our study, we found that a few hotspots of this species. Okay, I would like to say thank you for all the organization for this support. Okay, if you have a question, please contact me or find me around here. Okay, that's all from me. Thank you.
Any question? We've got time for a couple. No questions?
Okay, if no question, thank you everyone. Bye.
Well, hello everyone, good afternoon. Today I will be presenting to you some preliminary results for my master's dissertation thesis. When we talk about embryonic development in sharks, we can see that this is like the most diverse vertebrate when we talk about embryonic development, but we were talking about today about placentotrophy. What we know about placentotrophy is mainly because of the work of Hamlet. He worked with Rhizopronodon terranovae. And the present illustration, we can see the model that he proposed for placentotrophy. In this illustration, we can see the uterus, the attachment placement of the uterus, then the embryo is surrounded by a third membrane that in some cases is called egg case. And then we have the fetus placenta and the fetus. To give a little bit of context and history about this study, at the Universidade Federativa de Rio Grande do Sul in Brazil, is one of the oldest conducting collections in Latin America, and scientists have been conducting samples since the 1980s, especially of blue shark Rhinosa glauca, as this is the only species allowed to land in Rio Grande do Sul. So, since the 1980s, we have samples of different embryo stages, and this is the journey that I took for my master's. I'm going to describe all of this. To describe the embryonic development in blue shark, we made some microscopical inspections of uterus, placentas, attachment sites, and umbilical cords. We made some histological descriptions of placentas, attachment sites, and umbilical cords. And then we made some comparative morphology, mainly using the placentotrophy model made by Hamlet. In this plate, I present to you the second most complete embryonic development for placental sharks. We have a recently fecunded egg, a 2 millimeters embryo with which you can see a head, eyes, and gill openings already development. In picture number 3, we see the fins start to merge. In picture number 4, we see that the yolk sac start to differentiate in the placenta. The papery look part of the placenta is called the proximal placenta, and then the other one is the placenta that attached to the mother. In picture number 5, we have 2 embryos, one out of the uterus chamber and the other one within still in the uterus chamber, surrounded by histotrophy. And in picture number 6, we have histology of the attachment sites, in which we can see secretory cells and lipids. And here, this is just a almost full-term embryo. But something really interesting happens with blue shark. Otake and Misue, when they firstly described the embryos and the placenta for blue shark, they noticed that they do not have, or the embryos are not surrounded by a third membrane or the egg case. So when we started to open the embryos to make microstructural descriptions, we noticed that we started to find the egg case or the third membrane, the Otake and Misue, were missing. They were inside the embryos. So it seems that the embryos are eating this egg case, and we actually got the sizes in which they start to do so, because we found at certain points in development, egg case in the stomachs, and then on the intestine, or almost decomposed. This is not the first time that this happens, that this combination of methods occur in sharks, and it was firstly described by Gilmore for Carcariostatus. When we align the nutrition stages with growth, we can see that they group. I would like to thank the SCORE for financing my travel here, and all these Brazilian institutes that pay for the research.
We got time for one quick question.
Yeah, first of all, thanks for your talk, very interesting. I was just wondering if, because we know of interuterine cannibalism in sharks, right? So can it be the case that they're actually eating the eggshell of another shark that is in the uterus together with them and not their own eggshell?
In blue shark, when the egg is recently it's fecunded, or it's like, oh my God, sorry for my English, fecunded, the embryo, he goes through a process in which it attaches itself in a separate chamber, uterus chamber. So they actually separate from their sisters and brothers.
Okay.
Thank you.
Next up, we have David Abekasis with niche partitioning between sympatric species of skates.
Well, good afternoon, everyone. My name is David Abekasis. I'm a professor at the University of the Algarve, and I'm a researcher at CCMAR. These are both in Faro, Portugal, and niche partitioning is a central question in ecology, and it's also relevant for ecosystem-based management. And this study we're going to present now today, it's about where we tried to assess if the niche partitioning was taking place at the spatial, temporal, or dietary scale. So this study took place in the Luís Saldanha Marine Park, a small MPA located just south of Lisbon in Portugal, where we had identified the presence of 9 bathoid species, so skates and rays. And out of these 9, we decided to focus on 2 species, so the white skate and the thornback ray, mainly because these were the 2 most common species in the area, and also because they were present all year round and presented a high site fidelity to this area. Okay. What's going on? Okay, so for the spatial ecology, we used acoustic telemetry. We tagged 30 individuals of the white skate. We tracked their movements for around close to 2 years. Their average disc width was around 60 centimeters. We captured 50-50 in terms of sex ratio, and out of all of these individuals, only 2 were mature, 1 male and 1 female. For the thornback ray, we tagged 35 individuals. We tracked them for 2 years, more or less at the same time as the white skates. They were slightly smaller with an average disc width of 15 centimeters. There were many more males than females, and most of the individuals were mature already. Okay, so regarding the spatial ecology, in terms of home range areas, they presented very similar sizes, both in terms of KUD 95 and KUD 50, and we observed a very large overlap of their home range areas. Regarding their diel activity, both species were more active during the twilight and night periods, while staying deeper during the day. Regarding their diet, we looked into fecal DNA metabarcoding using 2 different markers, 18S for a more general perspective of their diet, and the 16S focusing on fish. And through this, we saw that the white skate had a broader and more diverse diet, whereas the thornback ray presented a more narrower trophic niche. We saw a clear clustering in the diets of these 2 species, indicating that there's a clear dietary differentiation between them. So to conclude, the coexistence of these 2 sympatric species is clearly driven by trophic partitioning and not at all by spatial or temporal segregation. And this study shows the importance of combining multiple methods to better understand the niche partitioning between sympatric species. Thank you.
We got time for a couple of questions. No questions? I've got one. What kind of sort of dietary differences do you suspect?
In terms of, so
trophic, you said trophic
yeah, so the white skates had a much broader trophic niche, so they fed on a lot different, on a wider range of prey species, whereas the thornback ray was more focused on specific species.
Okay, great.
Thank you.
Thank you very much. Next up, we have Miriam Zanforlin with thermal stress in reef shark neonates.
Thank you, Tom. So many awesome presentations about early life stages. We love our baby sharks. But unfortunately, Early life stages are the primary survival bottleneck across the animal kingdom. This is because they possess a set of traits that make them more vulnerable to mortality, unfortunately. And this is even more important for viperous elasmobranchs, as the embryos develop inside an egg case completely exposed to environmental conditions, and they develop off finite yolk reserves. After hatching, some species still possess internal yolk sac that help them making the switch to solid food, but if that first feeding event is delayed for any reason, starvation risk rises very quickly. So, unfortunately, whether and how an individual metabolism changing across the transition from the yolk to the prey has never been characterized in elasmobranch before. And that's what we wanted to do with epaulet sharks. It's a viperous species. It's very well characterized from a metabolic point of view. And the yolk to prey transition is very clean and traceable in the lab. So to do it, we used intermittent flow respirometry. It is a very popular method in physiology to estimate metabolic rate before and after a feeding event for the first meal and across the first month of feeding on exogenous food. And we wanted to quantify the specific dynamic action, which is essentially, is displayed in this very nice curve, and essentially is the response in the metabolism after you eat something, capturing the peak, the duration of the response, and the magnitude. And also we wanted to characterize and quantify the baseline metabolism in an individual just to staying alive. And we found very nice things, actually. So the first meal triggered an immediate and very strong metabolic response, corresponding to 1.7-fold in the peak, 1.7-fold increase in the peak above SMR within just 2.5 hours after the first meal. And this essentially is telling us that digestion is an immediate and substantial aerobic response and expense in hatchlings. Regarding the SDA metrics that I was talking about before, during the same period, we saw that the magnitude nearly tripled and the duration more than doubled. However, the peak metabolic intensity was essentially the same. So this tells us that there seems to be a fixed ceiling on how digestion can spike in terms of metabolism. So larger meals are accommodated by working longer and not necessarily harder. But we were also interested in understanding what was happening before the first feeding. And so we looked at SMR, so the baseline metabolism, and we found something very interesting. There was a positive, strong relationship with the latency in days between the yolk depletion and the first feeding event that corresponded to approximately a 3% SMR increase for each additional day of latency. However, we don't really know the direction of this casualty, meaning that whether delay in first feeding are rising metabolism up or individuals with higher resting metabolism just takes longer to feed for the first time. But what I can tell you today is that this window is a tightly constrained energetic framework. That beyond the lab means that the bottleneck that I was talking about before during this period is not really digesting competence, but is finding food. And this comes with ocean warming that we are experiencing today, will compress that pre-feeding window and will raise metabolic cost. And at the same time, reef degradation, prey mismatch will strip down the food that will end that delay. And for bending side attached species, well, they will converge in a very vulnerable life stage. Very interested in anything that concerns life history strategies and ontogenetic vulnerability, so find me later for any questions or anything else you want to talk about. Thank you.
Okay, we got no time for questions. Next speaker is Eswar Satyanarayana with the largest morphological and reproductive data set.
Hi Sharks, my name is Dr. Eswar Narayana. I'm from India. I work on east coast of India. And my topic is largest morphological and reproductive database of Pseudocarcharias comacari from eastern coast. So, can you identify this? Can you tell me the common name of this? Any guesses? By looking at that. This is crocodile shark. This species is the smallest living laminoid belonging to the Pseudocarcharidae, and it's found across the world. This is where I work. This is India, the Andhra Pradesh, Kakinada, and this is the landing center where I captured this. Here you can see the small crocodile sharks here. And the distribution is tropical and subtropical of Atlantic to Indian Ocean to Pacific Oceans. Caught as a bycatch and discarded often, and little is known for the history of this species. And most knowledgeable knowledge comes from the scattered records of very small size. So, from my landing surveys, I have collected the morphometrics of 247 individuals between 2022 to 2024. It is the largest data set from India. So, I am doing the morphometric collecting data from the landing center. So, few observations I have made through these 2 years. If you can see the bar, female landings are more in all the months, and during the August and February, the peak is more. And I also collected data on the gravid females. Those landings are also peak in the August month. And this suggests potential seasonal variation in reproductive occurrence. And size class distribution, most of the individuals are 80 to 99 centimeters range, and more gravid females were recorded in the August, suggesting the area may be important for the reproduction during this period. And these are the pups. So when they are just like processing it, and the pups came out from the this one, and like I have taken the morphometrics of this thing also. This study provides the first large-scale evidence from India indicating that the Bay of Bengal may be serve as a biologically important region for crocodile sharks, particularly in terms of reproduction. This study provides a baseline for future ecological and conservation studies. I would like to thank WCS India and RIST, and this is my WCS team, and I would like to acknowledge Rural India Supporting Trust, WCS India, a fisherman of Kakinada, who allowed me to do the morphometrics. Thank you so much. Any questions?
You got time for a question?
Yeah, please.
Hello. For catching the crocodile shark, how much they have to travel, the fishers, to go? Like how many hours or many days for catching it?
Actually, the fisher from Kakinada landing center, they have to go towards Bangladesh. Okay, so might be they travel around 12 days. The fishing days might be from 10 to 25 days. It's a bycatch actually. So there is no target catch, and we have only 6 records from India. This is the largest database. Thank you.
Okay, that concludes the first half of this session. There's now half an hour break.
Next up, we have Aurélie Benoit speaking about small rays, big threats, quantifying microplastics in the scaly whipway, whipray, Brevitrigon walga.
Hi everyone, this is Aurélie and I'm going to talk about my work on microplastics which I did as a part of my master's thesis. So here you see a landing center with lots of fishes and lots of business going on. On the other picture you see elasmobranchs and some other fishes. Now elasmobranchs are commonly landed as bycatch in the west coast of India and they often end up on our plates as a cheap source of protein. However, they carry something invisible within themselves, and that is nothing but microplastics. Microplastics are a global threat and have been documented in a lot of species and systems, but they remain fairly less studied in rays and sharks, especially stingrays. So I decided to look into microplastics from this beautiful species, the scaly whipray, scientifically known as the Brevitrigon walga, which is near threatened and vulnerable to pollution. So for this, I conducted my study in Malvan, which is a major fish landing center in the state of Maharashtra in India, and also a major tourism hotspot. So for this, I sampled during the pre-monsoon and the post-monsoon seasons, got the samples to the lab, digested it with KOH, filtered the sample, and then looked for microplastics under the microscope, and also did Raman spectroscopy to understand what is the polymeric composition of it. So what did we find? We found that all the sampled individuals contain microplastics, and the fibers are the most dominant ones, especially the transparent fibers, followed by the blue ones, and they are probably coming from discarded fishing gear like nets and ropes. We did not find any significant relationship between the body size of the fish and the amount of microplastics ingested. However, there's a striking result that says that microplastics are found in the largest concentration in the muscle tissue, followed by the gut tissue and then the gut content. This is probably because the microplastics are getting flushed out easily from the gut content and they are getting translocated to the muscle, and over time they are getting accumulated there, and that is what people love to eat. So that's a health concern. So there should be a better plastic waste monitoring and management system, and people should consume diverse seafood and not stick to species that are already vulnerable and on the risk of extinction. So yeah, thank you.
Thank you so much. We do have time for questions.
I have a quick one since there are no others.
Yeah.
I'm just wondering whether you were surprised that there wasn't more of a relationship with disc length, given that it's being accumulated in the muscle.
That could probably because my sample size was less. So maybe with a higher sample size, I can get something. Even I'm curious, why didn't I get anything? Because in the papers I've read, either that's a positive relationship, mostly, and there was one paper which had negative relationship, but even I'm curious.
Thank you very much.
Thank you.
All right, next up we have Stefany Rojas Perea telling us about Sentinels of the Estuary, how urbanization influences juvenile bonnethead shark health and nursery habitat quality.
Okay, testing. Perfect. Okay, good afternoon, everyone. My name is Stefany Rojas Perea, founder and biologist with the Salty Lab Research Collective. This project of ours in particular examines how urbanization may be influencing physiological stress in juvenile bonnethead sharks across Florida nursery habitats. More specifically, what we're asking is whether sharks in less disturbed systems exhibit different biological patterns than those in urban estuaries, and what that may mean on a broader scale for fisheries and coastal management. So, as many of you know, estuaries are critical nursery habitats for many coastal shark species, and they also support early survival. all these sharks, but they are also naturally variable environments where changes in salinity, oxygen, and water quality already place stress on the organisms that are here. Urbanization adds to this by introducing pollution, altering fresh water flow, and changing habitat structure. And together, these factors can actually create persistent environmental stress during early life stages. And as many of you know, these nursery habitats don't just support sharks, they also underpin coastal fisheries and local economies. So to explore this, we compared juvenile bonnethead sharks across urban and less disturbed nursery systems in Florida, which you can see here. And across these sites, we collected morphometric data alongside spatial context, allowing us to really begin evaluating how habitat conditions may relate to biological patterns. So when we examined size distribution across the sites, we observed a shift in structure. I do want to make a quick note that there is some overlap between groups, so these patterns should be interpreted as preliminary. The sharks from less disturbed systems tended to be larger, while individuals from the urban systems appeared smaller and more variable. Now, because body size in juveniles is linked to growth and survival, these trends are consistent with the possibility that habitat conditions may be influencing early life stage outcomes, because even subtle differences at the stage can actually represent early biological responses to environmental stress before larger scale changes even become visible. And as many of you also know, environmental stress in estuarine systems tends to build up over time and is often difficult to detect early, which leads me to why we chose bonnetheads for this study. They are very cute, but that's not exactly the reason we chose them. Bonnetheads show very strong site fidelity, meaning that they remain within these nursery habitats and can reflect local environment conditions. And if these patterns reflect differences in environmental stress, they suggest that nursery habitat quality is shaping early life stage outcomes with the implications for population stability over time. So the next phase of this project is going to focus on testing these patterns using physiological data. So we plan to integrate biomarkers such as glucose and lactate while standardizing sampling to also account for capture-related stress. We're also exploring non-invasive indicators such as mucus-based sampling. And then in parallel to that, because this also involves their habitats quite heavily, we're expanding habitat assessments through drone surveys and seagrass mapping, and incorporating stable isotope analysis to really evaluate the potential differences in diet across these sites. So all of that data will be paired with our spatial data sets to really strengthen habitat level comparisons, and overall this should create a pretty robust framework that can be applied across nursery systems for consistent monitoring. So just to quickly wrap, I really want to emphasize that the question here is not whether these environments are changing. We know that they're changing. What matters is trying to understand what those changes mean for ecosystems and the communities that are dependent on them. Because if we can detect stress early on, we have a better chance of protecting these nursery habitats and maintaining their long-term stability. And with the right scale, this approach does have the potential to support monitoring across many coastal habitats before decline even becomes visible. And with that, I'd like to say thank you. If you're interested in learning more about the work or potentially collaborating, please feel free to scan the QR code or talk to me after the talk. Thank you.
All right, we do have, right away, over there. A mic's coming to you.
Hi, how do you see this project being applied beyond bonnetheads?
Okay, excellent question. So the long-term goal for this project is actually not to just study one species, but to develop a framework for detecting early signs of habitat stress in coastal nursery systems. And like I mentioned, bonnetheads are a strong starting point because they actually, they show site fidelity and they remain really close to these estuarine habitats. And so as this project expands, we're going to be integrating the physiology, the habitat mapping, and the stable isotope analysis to really get a more robust framework. And it'll create an opportunity for larger scale monitoring, cross-system comparisons, and collaborative conservation efforts. It's really the goal to take this to more than just, Florida nursery habitats.
Thank you.
Thank you.
All right, next up we have Jared Cameron talking about environmental determinants of habitat use at Australia's only known sand tiger, gestation aggregation.
Thank you. Thanks everyone for coming. We know that ocean warming is a pervasive and accelerating threat to our sharks and rays. It's impacting their physiology and their distributions, and we desperately need a better understanding of how it's going to affect their reproduction. Of particular concern to me is the viability of critical reproductive habitats, and particularly when those are towards the warmer extent of distribution of a species, they may already be under threat. On the east coast of Australia, one of the species of sharks that we're concerned about is the sand tiger shark, or Carcharodon taurus, and this species has faced extensive population decline from extreme fishing pressure in the 1970s. It's been quite slow to recover because of their ecology, and they're also highly migratory and exhibit reproductive philopatry to only one known gestation aggregation in Australia, and that's Wolf Rock. Now, Wolf Rock is a really special place. It's used to some degree by about 16 or so different elasmobranch species, and some of our previous work went into getting it listed as an ISRA. But it is particularly important for our sand tigers. They spend the majority of their 9 to 12-month gestation at the site, and given that the site is located, again, towards the warmer extent of the distribution, they may be facing the highest temperatures than anywhere else across their range. So we were really interested in understanding how temperature drives residency and depth use for pregnant sharks when they're at Wolf Rock. And to do so, we employed acoustic telemetry. And there's been a long-term acoustic monitoring program at Wolf Rock since early 2010s. But recently, we implanted 15 females with temperature and pressure transmitters. And here you can see the mean number of detection days per month for our 2024 tagged sharks. They start in October because that's when the sharks were tagged. And you can see it increases through the spring and summer, but declines pretty rapidly in February. Now, what's interesting about this is this correlates with temperature. So we get the highest temperatures, both sea surface temperature in the red line, and the blue dashed line is our temperature at depth, which is catalogued by our receivers at about 20 meters. Another thing I want to point out here is that for most of the year, the blue line is much lower than the sea surface temperature. So the sharks have a bit more cool water that they can hang out in. But when we hit February, there's sort of this narrowing of the thermal profile. And that means that the sharks don't really have anywhere to go where they can retreat to the cooler depths. So where are they going when they leave the site? Well, if we look at the mean daily detection depth and the mean receiver temperature on those days, we can see that there's a correlation here where as things heat up, the sharks tend to move deeper to the point that they're no longer detected on our receivers. And this raises some really important questions about the viability of Wolf Rock in the future with climate-mediated ocean warming, because sand tigers may actually lose access to their only known gestation site in Australia as things heat up. If you're interested in learning a little bit more about this study, please do scan the QR code. There's also a video on there of a pup moving inside the stomach of one of the mothers at Wolf Rock, which is pretty awesome to see. And I'm interested in this problem globally as well. So if you are sort of looking into this area of climate change affecting reproductive habitats, then yeah, please do come find me and have a chat. Thank you.
All right, thanks, Jarrod. We do have time for a question, but can I please request those at posters to have quiet conversations and so we can all hear the speakers. But are there any questions for Jarrod? Okay, I'll ask a question.
Okay.
I'm sorry.
That's all right.
With the gestation, you said 9 to 12 months. Why the range, and is there anything that's driving it?
It has been narrowed down to about, we think it's about 11 months, but the range is just because there's so many studies out there that have looked at it and they've sort of quantified it differently. And it may be that there's sharks that spend more time at Wolf Rock and in those warmer waters that speed up embryonic development before they move down south to pup. So that could also explain the variation there as well. Yeah.
Thank you.
All right, thank you.
All right, our next speaker is Aurélie Benoit, talking about defenses down, reef shark immune system compromised during prolonged warming exposure.
Hi everyone, good afternoon. Thanks for introducing me. So today I would like to talk about my master thesis findings and how climate change can impact epaulet sharks' physiology. So I hope everyone knows that climate change is happening and it's threatening every species, including sharks. As ectoterms, they very much threaten. But unlike other fishes, sharks have a very slow mutation rate, meaning they are less susceptible to adapt to rapid environmental changes. If some species might be able to migrate away to cooler water, some, like for example, reef shark-associated species, just won't have that option, so they will need strategies and mechanisms to cope with global warming. So we already know that ocean warming is impacting reproduction and other biological function, but what we do not know is how the human system is going to be impacted. And yes, it is an important question as we expect bacteria to be increasing with rising temperatures. The elasmobranch human system is one of the most efficient among vertebrates. It's been refined over a billion years of evolutions, and they are known for their amazing healing capacity and low infection rate. Take these sharks, for example, who was capable of almost completely regenerating its dorsal fins within just a few months. This strong capacity is one of the reasons they've been around for so long. But ocean warming is not threatening these very traits. Recent work have shown that water temperature is directly impacting healing rates in newborn reef sharks. And even more concerning, scientists in Malaysia recently documented skin disease outbreaks that coincide directly with marine heat wave. While we could not prove that they were climate change driven, it's very likely, as we already know, that chronic stress suppresses human function across vertebrates. So this is the question becomes, what happens when sharks are exposed to prolonged warming? So first of all, I have to introduce you to my model species, the epaulet sharks. It's widely used as a model for conservation physiology for other reef-associated shark species. By understanding how they react to future conditions, this can give us a clue about how other species may or might not cope with future conditions. They also are reef-associated species, and we all know that coral reefs are among the most threatened ecosystems on the planet, and especially by ocean warming. We expected up to 5 degrees by the end of the centuries. But what that makes very challenging environment is that they're naturally dynamic. Reef flat temperature can fluctuate by 5 degrees or more within a single day. And this natural fluctuation means that reef organisms might already live close to the physiological limits. Add ocean warming on the top of that, and we're likely pushing many species beyond what they can tolerate. So with that in mind, we captured 24 epaulet sharks. We drove them 20 hours all up to the Australian Institute of Marine Science, where sharks were divided into 3 groups. A winter treatment representing end-of-century future winter conditions and incorporating daily fluctuation between 23 and 28 degrees Celsius. And that was to accurately reproduce what they're experiencing on the reef flats. Similarly, we have end-of-century summer groups. And finally, the last one was held static at 28 degrees, which allows us to understand the physiological cause of those thermal fluctuations specifically. And after 4 weeks of acclimation, blood samples were taken every 2 weeks at week 4, week 6, and week 8. And after that, we drove back the sharks 20 hours again and released them on the flat. So we started by using those very and widely used classic tools, hematocrit and hemoglobin. So what is standing out here on the result is basically nothing. We did not see many differences between groups or across week. And that was very surprising, and we did not really believe that. So we investigated a bit further, and I counted the total amount of blood cells. That took me a while, but that's when things start to be interesting, because after 4 weeks of exposure, we saw a drastic spike in the total blood cell concentration for sharks exposed to future summer conditions, and it was roughly 3 times higher than the other groups. And we found that this increase was driven by white blood cells, red blood cells, respectively suggesting metabolic strain and inflammation. But not only, we also found cells that we were not expecting to see in healthy sharks, dividing cell, abnormal forms, and also immature ones, and almost exclusively in the sharks exposed to future summer conditions. So this suggests a strong initial physiological stress response. The shark's body is essentially throwing every resource it has at the stressors, driving severe inflammation, but also oxidative stress. This is not a healthy response, and this is not a sustainable one. But you may already have noticed, things start to get interesting after 6 weeks of exposures. So 2 weeks later, the total cell count and abnormal cells return to a more acceptable values. So what does that mean? That might be a sign of potential thermal acclimation. It is genuinely exciting finding because it suggests that epaulet sharks may have physiological mechanism to cope with long-term ocean warming. But this plasticity comes at a cost, and to understand that cost, we need to look more closely at the white blood cell profile. So for you, I identified and quantified the different white blood cells, and that's when I lost my mind. But here above, you can see the key actors of the shark's immunity, and you can see they were strongly suppressed in the shark exposed to summer conditions. For example, the first one, the FEG, was not even barely here in the first place, where we have the CEG and the monocyte who are decreasing at the same time that the acclimation was occurring. And meanwhile, on the other side, we have less traditional immune cells who are increasing. For example, thrombocyte, whose primary role is coagulation, but who appears to have a role in immunity, really unclear, was really strongly recruited in sharks exposed to summer treatment. And this result altogether suggests that sharks exposed to future summer conditions have a complete reorganization of the immune system, with the main actor being suppressed, while other non-traditional immune cells are increasing as a potential compensatory mechanism. So what does that mean for shark conservation? So first, let's start with the good news. There is the fact that the total cell count and other stress markers normalized by week 6 and week 8 is one of the first signs of thermal acclimation in epaulet sharks. This is genuinely promising finding and suggests that at least some reef-associated species may have mechanism to cope with end-of-century conditions. But this acclimation comes at a cost, and one of these costs was the immune system. We saw a complete reorganization of the immune profile, and it is very unlikely that this immune profile will provide the same amount of protection against pathogens in the future. So this raises a real concern for future disease outbreaks, under ocean warming conditions, and not only for epaulet sharks, but for plenty of other shark species. And importantly, those are not hypothetical future conditions. Reef flat temperature are already reaching these extremes today, meaning this immune disruption might already be happening in the wild, and we have no idea. So this brings me to my final point. The classic tool completely missed this physiological stress and adjustments. This highlights the urgent need for shark-specific biomarkers and hematological data from wild population to have baselines, because without them, we're just simply blind, and we have no way to detect early signs of immune stress or anticipate these outbreaks before this becomes a conservation crisis. So I think I'm running a bit out of time, but that's just the thing that I would love to do in the futures, including, as I just say, identifying new biomarkers and baseline population. Just come back from the field where we brought them for the epaulet sharks. And also, I would like to investigate if there is potential vulnerabilities among life stages, including the babies, but also the pregnant sharks, as it might be a trade-off for reproduction. And finally, we only investigated ocean warming, but climate change is plenty of other stressors, as acidification and deoxygenation. So there's also a lot of work to do, and also potentially with fisheries and catch and release. So thank you so much for listening to me. If you have any question, I'm happy to answer it.
Thank you so much. We do have time for a question. This will be easier.
Thank you for the presentation. It was very nice work. But sorry if I misunderstood. Didn't you use a control group, like having the same temperature?
The winter group was quite of a control group because it's what is happening on the reef at the moment for like springs for them. So that was our control group.
Okay, perfect.
Okay, great. Thank you so much, really. Next up, we have Gary Ostrander speaking on reevaluating cancer incidence in chondrichthyans, empirical evidence, misinformation dynamics, and the shark cartilage fallacy.
Thank you. Good afternoon. I appreciate you coming to my presentation today. I realize it's different than the presentations that most of you will be attending this week. Today I'm going to be talking about the status of chondrichthyan cancer, and particularly related to the myths around shark cartilage's medicinal properties. Little background to get us started. The Any cancer, whether it be in a fish or in a human, begins with a single cell that clonally expands. As the tumor begins to increase in size, it needs to develop a blood supply to bring in nutrients and to eliminate waste. This process is called angiogenesis, and the developing tumor secretes factors that stimulate angiogenesis. Work at MIT quite a while ago demonstrated with bovine cartilage that there were apparently factors that promoted angiogenesis. Bovine cartilage was difficult to get. The researchers were in Boston, so they went down to the docks and they picked up shark cartilage. Shark cartilage also showed or demonstrated, it appeared to show a slowing or in angiogenesis. This whole idea then was perverted and corrupted, and I use those words I think appropriately, as you'll see, by a fellow named William Lane in the 1990s. He put out a book saying that sharks don't get cancer and followed it with a second one. His premise was that since sharks have a skeleton entirely made of cartilage, they would be immune from cancer. Of course, that's not true. He argued that the anti-angiogenic factors in cartilage were what was responsible for doing that. He further went on to argue that if you ingest shark cartilage, you can cure cancer or treat cancer, and if you don't have cancer, you should take shark cartilage. This is the premise of his work. He has no background in medicine, no experience in those areas. He ran a company called Cartilage Consultants, and he worked with Lane Labs, which was a company run by his son, and the two of them promoted therapy using shark cartilage products, and they've been the leading promoters of products like this since the 1990s. Now, Lane was a marketer, and what he realized is he needed clinical trials, and he found that he could not do clinical trials in the United States or in Europe because the standards for doing a clinical trial on humans with a drug or a compound requires that there be some demonstrated efficacy. So he turned to emerging nations, and he did these trials in a number of emerging nations. The first was in Cuba. He claims to have treated 29 women with advanced breast cancer, and the treatment was a 16-week course of retention enemas with shark cartilage and drinking fruit juice with shark cartilage. 9 women died during the treatment, and 6 more died within a few weeks of completing the treatment. He claimed a couple of years later that the remaining 14 were all alive and well and cured of their cancers. His colleague said differently, they had all passed. It is significant that the 5-year survival rate for stage 3 breast cancer is about 50% with no treatment whatsoever. Anytime you see an advertisement that suggests one drug will cure multiple cancers, you need to be suspect, because every cancer is different. Breast cancer is different from lung, and it's different from prostate, and the treatment is different. And when those same arguments say you can treat other diseases, you're certainly suspect. Over the years I've been investigating this or following this, I've looked at a lot of claims for medicinal applications of shark cartilage. These are primarily found on the web, tied to products that are being sold, found in magazine ads, product inserts. What's significant about all of these maladies is there's not been a single peer-reviewed clinical trial to show that shark cartilage works for any of them, including all of Lane's studies on cancer. Finally, in the late 1990s, it was determined that they needed to go ahead and do some human clinical trials. First peer-reviewed paper that came out on this was a study that involved 60 patients with a variety of different types of cancer. Well, at one point it looked like about 10 of them there was some stabilization of the growth of their tumors. By 46 weeks after the end of the treatment, all 60 patients were exhibiting tumor progression. The researchers peer review published this, and they concluded that shark cartilage basically did nothing. Subsequent studies were done in the United States into the early 2000s and across Europe with the same result. Sharks do get cancer. To date, there's been over 60 examples that have been reported and found their way into the literature, benign and malignant, and even a couple of examples of chondromas. The next 4 slides, I don't expect you to spend a lot of time on. I'm certainly not going to. I just want to give you a sense for the breadth of the species that exhibit cancers. Maybe some of the ones you work on are in there. And I'll also point out that a number of these sharks are being collected from zoos and aquaria, which has worked out really well because when these animals have been on exhibit for 5 or 10 years, and then they present with the cancer, there's usually a vet there that can accurately diagnose it, collect tissue samples, and they often publish this work. Sharks, skates, rays, chimerids, organisms more primitive than sharks, organisms more advanced than sharks, they all get cancer. This is a chondroma from a dogfish shark that was collected in the North Atlantic on a fishing expedition. This is a hypodermal lipoma that was found in a swell shark. This is a cutaneous fibroma that was collected near Hawaii on a tiger shark. 2 pathology slides, only 2. A and B are significant because these are 2 very different tumors. The one on the left is a cholangiocellular carcinoma, and the one on the right is a mesothelioma. Anytime you have 2 different primary cancers in a single organism, in this case it was a blue shark, 1 animal, you suspect the animals have a predisposition to cancer or they've encountered a significant carcinogen exposure. This is not uncommon in sharks. Panel C is a neuroblastoma from a ratfish, and panel D is another shark that exhibited multiple tumors. This one is a follicular follicular lymphoma from a sandbar shark. Last slide on the pathology. On the left is a renal cell carcinoma. The middle panel on the left shows you the poorly differentiated area that's so characteristic of this tumor. And the bottom panel shows you mitotic figures, which tell you it's a rapidly fast-growing tumor. The tumor on the right is one of the chondromas I mentioned. Panels E and F are showing you higher magnification of the chondrocytes with the immature cartilage development. Sharks, there is no evidence at all to suggest that sharks present with cancer at rates lower than any other animals. Sick and compromised animals are likely eliminated from the population by predation, and there have been no systematic tumor surveys of sharks to assess, or rays or skates to assess the incidence of cancer. There is also no good methods for reporting examples of cancer in these animals by fishers who see them. So at this point, all we can say is sharks exhibit cancer at unknown rates, and as the years go by, more and more reports are coming out, though I'm not suggesting that there's any kind of increase in the rate. So I'll conclude by pointing out that pseudoscience and really the failure of our education systems to teach critical thinking skills around the internet are leading to the pervasiveness about myths about shark cartilage. The reality is millions and millions of dollars of this product is being sold every year, and individuals with limited time and limited resources are buying it and ingesting it, hoping it will deal with whatever ailments they have, especially with cancers. Likewise, major corporations are able to sell shark cartilage worldwide with no evidence or data to support its efficacy. As long as these trends continue, there is going to be targeted thinning of sharks for the shark cartilage industry, and it's going to continue to exert downward pressure on ecosystems. Thank you.
Thank you for an interesting topic. Any questions? We have time for one.
Thank you for that. It was great. Just wanted to think about that last question or that last comment that you made about the fact that there hasn't been any targeted studies on this. How would you suggest going about that? Because it's quite random, right?
Exactly, and I am not going to be an advocate for going out and sacrificing 1,000, 2,000 sharks in an area or a broad area to assay for tumor development. It's just not a good thing for the ecosystem. What has happened, and if you were looking at the slides really closely, you'll notice that there were a lot of melanomas in the skates and rays, and that was all because one fella in Ireland was going down to the docks religiously asking the fishers for examples of tumors in skates and rays. So probably what you do, and it's a great question, is you would work out with observer programs or fishing programs for, and maybe incentivize the fishers to look for and report back examples that are seen. Thank you.
Thank you so much. And he is speaking on the conservation of ancient relationships, chondrichthyans and their parasites.
Thank you. So, one might say, best for last, let's talk about parasites. What you see here is the endangered shortfin mako. We all know that. What I see here is the shortfin mako plus 37 potentially endangered species. What you see here is the taupe shark, a critically endangered species. What I see is a taupe shark plus 48 potentially critically endangered species. You see where this is going. I can continue like that for the next hour, but I think that would be a bit dull. So let's just one more time. This here, the critically endangered scalloped hammerhead. I see the scalloped hammerhead plus 56 potentially endangered or critically endangered species. Now, the chondrichthyan body serves as a macrohabitat for a large variety of internal and external organisms, symbiotic in nature, mostly parasites. And you can see here all those different groups, some of them with a very high diversity, others with a relatively limited diversity in chondrichthyans. These three groups here, the tapeworms or cestodes, the monogenians, and the copepods, they represent up to 85% of the entire chondrichthyan parasite fauna. So there is a lot if we just look close enough. And especially the tapeworms, the cestodes, they are highly diversified in this host group. Together with their chondrichthyan hosts, they form the most ancient host parasite system that we know of. So their co-evolution dates back about 270 million years, so to the Permian period, where we have found shark coprolites with viable tapeworm eggs inside. And of course, during this long, extended time of co-evolution, you might expect that there have been vast diversification events in the cestode clade. Now, this here is the phylogeny of tapeworms, and you can see here that the earliest branch of tapeworms, the Gyrocotylidians, which is the most basal groups, they occur in chimeras. And along the way down to the most recent forms, we have now 10 out of 19 orders of cestodes that exclusively infect chondrichthyan hosts. Right now, we know of 1,200 species, slightly over that, so roughly the same amount as potential host species out there. However, we estimate that the true cestode diversity ranges more or above 5,000 species. So there is still a lot to be discovered. And this group in particular, I would say, or I would argue that we currently do not know whether those are really parasites or commensals or mutualists, so the boundaries are not very strict. In fact, we have to be very careful when we place the name parasite on an organism. So over here you can see cirripedes infecting a Atmopterus species. This species is clearly affected by the parasite load, and it's probably not very pleasant to have those cirripedes sticking out of your cranium, right? However, if we look at the right side, this is what I would consider a perfectly healthy stingray spiral intestine to look like. In fact, if you open up a stingray, or a shark for that matter, and you look inside the spiral intestine and you do not see any tapeworms, something might be severely wrong in the ecosystem. Or even worse, something might be severely wrong with the shark individual or the ray individual. We all know that constrictians are a highly threatened host group right now. Well, host group, you see who's talking, a parasitologist. A highly threatened organismal group right now, far more threatened than, for instance, mammals or reptiles or birds, but that is nothing in comparison to the symbiont assemblages that they carry. So at this stage, we estimate that around 10 to 18,000 species occur in constrictians alone. That represents about 8 to 15 species per available host species. And the extinction, or in that case, co-extinction risk might range anywhere between 2,300 and 5,700 species. Now, this is a very conservative number. I would estimate that the number is more likely between 5,000 and 10,000 species. So that's a shocking scenario, obviously. The big question is, why should we care? Well, I certainly do. Well, parasites are responsible for about 70% of all trophic links in any given food web. They also represent about 40% or even more of global biodiversity, and you can just imagine what would happen if they wouldn't be there. Parasites are known to be the ecosystem engineers. So in this case, they have very important functions in any given ecosystem for the health and the diversity. And over here, I list only very few positive attributes that parasites might give to ecosystems, but not only that, but also to host populations, host species, and even host individuals. For instance, they bioaccumulate toxic substances and thereby keep the host alive. Now, co-extinctions are a real threat, and we estimate that they represent the most common form of biodiversity loss to date. About 30% of all our symbiont species are currently threatened. I think this is also a very conservative number, and that's most likely due to their very high host specificity or very complex life cycles. Now, of course, if those species are missing from the ecological theater, there are major downstream consequences that we don't even know of or hardly understand. But we humans, we have a very funny approach to conservation. We basically select the species based on the likability or based on the attractiveness that species has to us humans, and that's the wrong approach. Invertebrates, for instance, most parasites, they are certainly not included in any conservation efforts. They haven't been a conservation priority nor conservation targets. And yeah, if we ignore them, we basically ignore the conservation status of a majority of life on Earth. So we are faced with a very paradoxical situation right now. So we have to perceive the symbiont assemblages and the constrictians as ecological units and try to protect them. We have to actively promote the protection of constrictian and parasite systems. And this is what we are currently doing with the establishment of the IUCN SSC Parasite Specialist Group. If you would like to know more about the IUCN SSC Parasite Specialist Group, please reach out and remember the Wi-Fi password, race rule. Well, it should be parasites rule more. Thank you.
Thank you so much. I will not have any questions. There's no time for questions, sorry. And people can come up to you because you are the last of the session. So please ask all the interesting stuff about parasites. And please join me in thanking all of our speakers for this session. Thank you, everyone.